Very happy afternoon, students. I hope you are all safe and sound at home. Once again, welcome on behalf of RGR Academy. So we'll start with the bio session today. As we discussed yesterday, we began with a new unit, that is cell biology. We started with cell, the structure and functions. There we discussed mainly about the cell. The introduction about the cell that we discussed, we classified basically the cell biology into the outer membrane. Outer membrane, we discussed about the cell wall and cell membrane. And coming to the inner part, the fluid matrix, the part that we call as protoplasm. Under protoplasm, we discussed about nucleoplasm and cytoplasm. And coming to the cell organelles, right? So we begin with the cell organelles. Now we'll discuss about the cell organelles, what all the organelles which present inside the cytoplasm that we're going to start for today. All right. So now, without wasting our time, let us begin with today's session. So now begin with the mitochondria. First, we'll start with the mitochondria. So I hope you recollect what all things that we discussed, especially with the nucleus is concerned. When we come to nucleus, we discussed about the structure of the nuclear envelope, the nuclear pore, and we discussed about the chromatin. Yes, of course, and we differentiate the chromatin as euchromatin and heterochromatin. And we've seen the 6 to 7 different differences between euchromatin and heterochromatin. So I hope you followed that as well. So, and we discussed about the chromosome, how the chromosomes were made up of, and what is a gene and what is allele, and also we discussed about the karyotyping. So how the karyotyping is beneficial, so what are the advantages of karyotyping. So all these things we discussed based upon the zygiogram karyotypic technique. How do we group this chromosomes? That 7 groups that also we discussed. So this is what actually we have done yesterday. So now we'll continue with this chapter, the cell biology. So hopefully we try to complete this chapter along with this cell, the unit of life. And additionally, we're going to look into the microscopy also, because the microscopy, the tools that used to look into the cells and the internal structure of the cell. So what are the different, the latest techniques that we have, like flow cytometry, the spectroscopy, little bit of additional information also that we're going to discuss in this particular chapter. Once we're done with this chapter, if time permits, we'll start with the next chapter, that is your cell cycle. So cell cycle, we'll try with the cell cycle. If again the time it's okay for us, then we go with the mitosis and then meiosis. If not, we continue with mitosis and meiosis tomorrow along with the biomolecules. Now we'll start with mitochondria. We move on to the cell organelles, continuation of the cell, the unit of life. Fine. So coming to the mitochondria. As far as the mitochondria is concerned, so the mitochondria is actually the thread-like granule which presents. When you talk about the mitochondria, the things which comes in your mind is actually mitochondria means it's thread-like granule. So let me reduce the size. Okay, thread-like granule. So the thread-like granule is actually the meaning for mitochondrium. So thread-like granule. Coming to this, actually, so usually we call the mitochondria, the nickname that we give for mitochondria. So mitochondria, usually we call it as a powerhouse of the cell. So this is called as powerhouse. So can you tell me the reason why it's called powerhouse of the cell? So it's a very common question. Which of the cell organelle is called as powerhouse of the cell? The mitochondria is called as the powerhouse of the cell. So can you tell me one particular reason why we usually identify this particular cell organelle, mitochondria, as the powerhouse of the cell? Fine. So coming to the mitochondria, the name actually is given by Benda. The mitochondria, the name which is coined by a scientist named Benda. And when coming to the discovery part, so the name is coined by Benda. And coming to the discovery part, who discovered this particular? Can someone tell me who discovered mitochondria? This is mitochondria. The name is coined by Benda. Who discovered mitochondria? What's the name of the scientist who discovered mitochondria? You said it's correct, produce ATP, so adenosine triphosphate. It produces that's energy currency of the cell. So since it's energy currency of the cells, it produces the ATP. So this we call it as the powerhouse of the cell. So coming to the discovery part, this was actually first identified by Kolliker along with Richard. It was first discovered by Richard Altman. Richard Altman in the year 1890. So even before the colica, you could see a person named Richard Altman, and then only becomes like colica. So the Richard Altman was first discovered after the Richard Altman, only the confirmation of the mitochondria, which was made by the colica. The first credit goes to the person named Altman. So if you look at the options, the question comes who discovered. If you could see this particular person's name, Richard Altman, then you go with this. If this Richard Altman is not there, if the colica option is given, you just go with the colica, right? But both the names will be given. First priority you need to give for the Richard Altman. So it is actually scientifically proven he is a person who first discovered mitochondria. Fine. So this is about mitochondria. And coming to the chemical composition, when it comes to the chemical composition of mitochondria, this consists of 70% of protein, 70% of protein, and this consists of lipid of 25 to 30%. The chemical composition of the mitochondria is 70% of protein and lipid, which comes 25 to 30%. From this 25 to 30 percentage, this 25 to 30 percentage, the 90 percentage of the lipids, which is actually made up of phospholipids. What type of lipids? These are all phospholipids. This 25 to 30 percentage, in which majority of the lipids, this consists of the phospholipid. So 90 percentage, which consists of the phospholipid, and the remaining 10 percentage, right? The remaining 10 percentage usually, which is of cholesterol. Usually the mitochondria, which consists of cholesterol, vitamin E, and carotenoids. This consists of cholesterol, vitamin E, and carotenoids. So these are all like the additional, the remaining 10%, but majority as far as lipid is concerned, this consists of phospholipid. This consists of phospholipid. Fine. The remaining 10% of cholesterol, vitamin E, and the carotenoids. And one more thing, as far as mitochondria is concerned, mitochondria is usually pleomorphic in nature. What do you mean by pleomorphic? So what is pleomorphic? So this is a pleomorphic in nature. So what do you mean by pleomorphic? Along with this, the structure, when you come to the structure of the mitochondria is concerned, we have the outermost membrane. This is the outermost membrane, and we have the inner membrane. This part is the inner membrane, and the space in between the outer membrane and the inner membrane, this we call it as the inner membrane space. And the inner membrane, you could see the matrix, and the matrix which consists of the mitochondrial DNA along with the ribosomes. This consists of mitochondrial DNA and the ribosomes, and also this consists of this cristae part that you could see, right? This cristae part, which consists of a specialized region. This specialized region is called it as oxysomes. Can someone tell me like what are oxysomes? What is oxysomes? What is this axisome? Oxysomes. So these oxysomes, what is actually like the function of the oxysome? The cristae part. The cristae part that you could see, right? Here this consists of this oxysome. You could see the black dots here, right? These black dots, indeed, we call it as oxysomes. Okay. So what is the main function of oxysomes, and what do you mean by pleomorphic? Pleomorphic means like it doesn't have the regular, a proper shape. It keeps on like changing its shape. So we call it as a pleomorphic. So mitochondria is one of the cell organelles which won't be having a clear shape, like it keep on changing its shape and structure. So we call it as pleomorphic. Okay, this is cell biology. We're talking about the cell, the basic unit of life. We're talking about the cell, basic unit of life, or cell biology. This chapter we're dealing with. So oxysomes. These oxysomes which present on this cristae, region, and this oxysomes, which is otherwise called as ATP synthase or elementary particle. This is otherwise called as ATP synthase. This is called as ATP synthase or otherwise called as elementary particle. So from this part you can get a question. And this oxysomes which consist of FO, F1 region. So this consists of FO, F1. This consists of FO and F1. What is the main function of this FO, F1? This FO, F1 only we call it as the different name that we're giving. FO, F1 or oxysomes, or this is called as ATP synthase, or this is actually even called it as an elementary particle. Now, what is the main function? Main functions of this FO, F1 particle, or what is the main function of this oxysomes? Can someone tell me? Again, the answer is like ATP. So synthesis of ATP is the main function of these oxysomes. ATP synthase or elementary or FO, F1, whichever the parts. If the question they ask, the answer is ATP synthesis. So this is what the answer that you can give related with all the four different names. So this is a release of ATP. So this is what you could see in the crystal cristae region. you could see these substances which we call it as an elementary particles. Coming to the matrix part which consists of the ribosomes and the mitochondria DNA. So this matrix part which consists of the ribosomes and the DNA. Coming to the matrix part, this consists of 70% of enzyme. The matrix part which consists of 70% of enzymes. So what for the 70% of enzyme? Because the 70% of enzymes which is responsible for Krebs cycle, electron transport chain, these enzymes which are responsible for Krebs cycle, electron transport chain, oxidative phosphorylation. So those are the important functions where this 70%, the 70% of enzyme which present in the matrix region. Where these enzymes are present? These present in the matrix region, and these enzymes, the matrix region which consists of the enzymes, right, along with the mitochondria and mitochondrial DNA and ribosome, this also consists of enzymes. These enzymes is playing very important role in the Krebs cycle. ETC means electron transport chain, and also for the oxidative phosphorylation. So oxidative phosphorylation. And these oxidative phosphorylation, these are like an enzymes which present in the cristae region. Where exactly in the matrix part in the cristae region, you could see all these enzymes which is present. And one more important thing where the mitochondria we're talking about, right? Where this mitochondria, which should not be like depending upon the nuclear DNA, because it's having its own DNA. So mitochondria it's having its own DNA, the mitochondrial DNA. So it need not depend upon like all the proteins and everything has to be produced from the nucleus or nuclear DNA. So mitochondria, since it's having its own DNA, so it can produce. So you would have studied about the central dogma. So this replication, transcription, and translation, this together, this process we call it as central dogma. This is what actually the DNA which is performing. What is the replication? Conversion of parental DNA to the daughter DNA. What is the transcription? The daughter DNA which is getting converted to RNA. What is translation? Where the RNA is getting the coordinated effects of RNA, mRNA, and tRNA together it produce a protein. This is called as translation. So replication, transcription, translation, இந்த மூணுமே சேர்ந்ததுதான் what we call it as central dogma அப்படின்னு சொல்லுவோம். So இந்த process நடக்கணும்னா nucleus, the nuclear DNA is very important. But mitochondria it cannot depend on the nuclear DNA at all the time where it requires for protein. ஏன் அப்படின்னு reason கேட்டீங்கன்னா mitochondria இந்த ஒரு cell organelle மட்டும் it's having its own mitochondrial DNA. So I told you it's having its own DNA. So this DNA will take care of all the requirement of the mitochondria of all the proteins that is required. So அதுக்கு வேண்டிய protein எல்லாமே requirement யாரே fulfill பண்ணிப்பாங்க கேட்டா மைட்டோகாண்ட்ரியாவே ஃபுல்ஃபில் பண்ணிக்கும். ரீசன் என்ன அப்படின்னு கேட்டீங்கன்னா மைட்டோகாண்ட்ரியாவுக்கு அதுக்கு அதோட டிஎன்ஏவே உள்ள இருக்கு. இந்த டிஎன்ஏ தான் மைட்டோகாண்ட்ரியல் டிஎன்ஏன்னு சொல்லுவோம். ஸோ இது எதை டிபெண்ட் பண்ணி இருக்காது அப்படின்னு கேட்டீங்கன்னா எதை டிபெண்ட் பண்ணி இருக்க வேண்டிய அவசியம் இல்லை. மெயின் நியூக்ளியர் டிஎன்ஏ டிபெண்ட் பண்ணி இருக்க வேண்டிய அவசியம். இது ஒன் ஆஃப் த மெயின் ஃபீச்சர் தட் யூ குட் see in case of mitochondria. Not only producing energy. நான் சொன்ன உங்களுக்கு ஆக்சுவலி பிளியோமார்பிக் அப்படின்னா என்னன்னா it keep on changing the shape, it keep on changing, ஒரு ஃபார்ம்ல இருக்காது. சரிங்களா? அது ஒரு வைரஸ், நீங்க வைரல் பார்ட்டிகல்ஸ் சைஸ் WBC எல்லாம் எடுத்துக்கிறீங்க, நியூட்ரோஃபில்ஸ் எடுத்துக்கிறீங்க அப்படின்னா அது ஷேப் சேஞ்ச். சில நிறைய வைரல் டிசீசஸ் இன்னைக்கு வரைக்கும் வேக்சின் கண்டுபிடிக்க முடியல. ஒன் ஆஃப் த மெயின் ரீசன் சயின்டிஸ்ட் எதுக்காக வேக்சின் கண்டுபிடிக்க முடியலன்னா இன்னைக்கு ஒரு ஷேப்ல இருக்கும், அது வேக்சின் கண்டுபிடிச்ச பிறகு அது வேற ஷேப்புக்கு மாறிடும். So this property is called as pleomorphic. That is what even you could see in case of mitochondria. மைட்டோகாண்ட்ரியாக்கு you will not see the proper shape. இது ஷேப்பை மாத்திக்கிட்டே இருக்கிறதுனால, ஸ்ட்ரக்சர் மாத்துறதுனால, this we call it as a pleomorphic. Okay, so ribosomes, of course, it is present. So you do have its own the ribosomes which present inside the mitochondria. Yes, of course, it's having its own ribosome. Oxysomes தான் என்ன சொல்றேன்னா கிறிஸ்டே பார்ட்ல இருக்கிற ஸ்பெஷல் FO-F1 பார்ட்டிகல்ஸ் பேர் தான் ஆக்சிசோம்ஸ். இந்த ஆக்சிசோம்ஸ் இன்னொரு பேர் நான் கொடுத்திருக்கிறது ATP சிந்தேஸ் சொல்லலாம், எலிமெண்டரி பார்ட்டிகல்ஸ் சொல்லலாம், இல்ல FO-F1 சொல்லலாம். This oxysomes which plays a very important role in the synthesis or release of ATP. Because main function of the mitochondria function it's a powerhouse for synthesis of ATP. இது யார் ப்ரொடியூஸ் பண்ற மைட்டோகாண்ட்ரியாலன்னு கேட்டா ஆக்சிசோம்ஸ் and the particular structure FO-F1 என்ற ஸ்ட்ரக்சர்ஸ் தான் ப்ரொடியூஸ் பண்ணுது. சோ இது எங்க இருக்குன்னு கேட்டீங்கன்னா கிறிஸ்டே என்ற ரீஜன்ல இருக்கு. So this is what the function of what is that called oxysomes. Okay, so ribosomes, suppose like 70 as ribosomes that you could see. So and of course being differentiated the ribosomes in the prokaryotic 70 as eukaryotic you could see like 80 as ribosomes that you could see. So this is about Central dogma is a very simple process. எதுக்கு நம்ம central dogma நம்ம சொல்லுவோம் அப்படின்னு கேட்டீங்கன்னா, central dogmaல வந்து replication, transcription, translation, ஒரு செல்லுக்குள்ள இருக்கிற DNA என்ன ஆகணும்னா புரோட்டீனா மாறணும். கடைசி நமக்கு என்ன ப்ராடக்ட் நமக்கு we are supposed to get only the protein. அந்த புரோட்டீன் நமக்கு எப்படி கிடைக்குது அப்படின்னு கேட்டீங்கன்னா because of the central dogma process. Central dogma processன்றது என்ன? ரெப்ளிகேஷன், டிரான்ஸ்கிரிப்ஷன், டிரான்ஸ்லேஷன் மூணுமே சேர்ந்ததுதான் உங்களுக்கு சென்ட்ரல் டாக்மான்னு உங்களுக்கு சொல்றோம். FOF1 வந்து இன்னொரு பேர் ஆக்சிசோமுக்கு வந்து அது பார்ட்டிகல், the particles which present in the crystal. ஆக்சிசோமோட இன்னொரு பேரை தான் நம்ம FOF1, this is particles which present. The other name for oxysome, FOF1 or ATP synthesis கூட சொல்லலாம். So coming for we ask for the central dogma. உங்களுக்கு central dogma பத்தி கேட்கறதுனால I just explain what is the central dogma because we're talking about the cell. Central dogma அப்படின்றது என்னன்னு கேட்டீங்கன்னா, what is the central dogma? Central dogma is the complete process. We call it as replication, transcription, then translation. Together, these three processes combined together, we call it as what? We call it as a central dogma. So what is replication? Replication is the parental DNA. So parental DNA is getting converted to the daughter DNA. So DNA is helical strand, இல்லையா? நம்ம DNA வந்து helical strandல இருக்கும். So இது ரெண்டுமே parental. The parental DNA என்னவா கன்வெர்ட் ஆகும்னு கேட்டீங்கன்னா daughter DNA வா ஒரு காப்பி வந்து daughter DNA வா மாறும். So this is called as replication. And produce on a daughter DNA ல இருந்து என்ன ப்ரொடியூஸ் ஆகும்னு கேட்டீங்கன்னா you could see the formation of messenger RNA, mRNA form ஆகும். This process is called as transcription. Then mRNA வந்து என்னவா ஆகும்னு கேட்டீங்கன்னா mRNA along with the tRNA and rRNA. So tRNA இங்க இருக்கும், coordinated effect of tRNA and rRNA, and this process is called as translation where you could see the formation of protein. So this is otherwise called as protein synthesis. So together ஒரு DNA வந்து கடைசியா வந்து புரோட்டீன் எப்படி ஃபார்ம் ஆகுது, இந்த ப்ராசஸ்க்கு பேரு தான் நம்ம என்ன சொல்லுவோம்னு கேட்டீங்கன்னா we call it as a central dogma. இதுல சில எக்ஸெப்ஷன் வரும். ரூல் என்ன சொல்றதுன்னு கேட்டீங்கன்னா எப்பவுமே டிஎன்ஏல இருந்தா என்ன ப்ரொடியூஸ் ஆகணும், RNA ப்ரொடியூஸ் ஆகணும். பட் சில கேசஸ்ல என்ன ஆகும்னா RNA வந்து டிஎன்ஏவா மாறும். Getting what? There are certain exceptions where you could see RNA is getting converted, but the rule central dogma says that DNA always gets converted to RNA. That is a transcription. So where and which exceptions, which are the cases where you could see there is a conversion like reverse mechanism which occurs where RNA again go backslide and get back to DNA. Can you tell me what is this process called and where and all you could see this type of process? அப்போ what is it called? RNA வந்து DNA வா மாறுது. So RNA வந்து DNA வா மாறுதுன்னா what is the process called? But rule says the central dogma இதுதான் flow. Parental DNA converted to daughter DNA. Daughter DNA gets converted to like RNA. RNA வந்து என்னவா ஆகணும்னு கேட்டீங்கன்னா புரோட்டீனா மாறணும், ரைட்டா? சோ இந்த ப்ராசஸ் வந்து சென்ட்ரல் டாக்மா, நார்மல் ப்ராசஸ் நடக்கிற ப்ராசஸ். சில இடத்துல வந்து ரிவர்ஸ்ல நடக்கும். எது? டிரான்ஸ்கிரிப்ஷன் ரிவர்ஸ்ல நடக்கும். அப்ப என்ன ஆகும்னு கேட்டீங்கன்னா where you could see this is getting converted, the RNA is getting converted back to what? Converted back to the DNA. So this process called as reverse transcription. This process called as reverse transcription. இது எங்க நடக்கும் அப்படின்னு கேட்டீங்கன்னா mostly in case of retrovirus. Right? So the retrovirus like HIV virus எல்லாம் this type of process which occurs by means of an enzyme called as reverse transcriptase. Yes, you're correct. Reverse transcriptase வந்து என்ன பண்ணும் அப்படின்னு கேட்டீங்கன்னா RNA வந்து DNA வா மாத்தி விடும். So ரிவர்ஸ்ல கொண்டு போகும். So இந்த ப்ராசஸ் வந்து சில வைரல் குரூப்ல எஸ்பெஷலி ரெட்ரோ வைரஸ்ல இந்த மாதிரி HIV வைரஸ்ல எல்லாம் வந்து நீ என்ன பண்ணுவீங்க பாப்பீங்கன்னு கேட்டா இந்த மாதிரி ப்ராசஸ் நடக்கும். இந்த ப்ராசஸ்க்கு பேரு reverse transcription. The enzyme which is responsible for this particular process called as reverse transcriptase. Clear up? So this is about the central dogma. Right? So mostly sometimes in the RNA interference la kuda the biotechnology part la kuda idha pathi inga padichirupinga. So when we come to that particular chapter we'll be discussing in detail about the central dogma. Since you asked the question like what is the central dogma, I just explained this process. So now I hope you are clear with the central dogma. In the process வந்து nuclear DNA ல நடக்கும், நியூக்ளியஸ்ல நடக்கும். ஆனா மைட்டோகாண்ட்ரியாக்கு வந்து என்ன டிபெண்டன்சி கிடையாதுன்னா it may not depend on the DNA, that is like nuclear DNA for the protein synthesis for all the time. Yeah, the reason is because of mitochondria. This cellular organelle, this Vacuoles has got its own mitochondrial DNA, so it need not depend on the nuclear DNA for the protein. It can manufacture its own protein because it's having its own mitochondrial DNA. அதனால நீ நியூக்ளியஸ்ல மட்டும் தான் DNA இருக்குமான்றது கிடையாது. So mitochondria இந்த organelle கூட it's having its own DNA, so it can fulfill its needs for the production of any protein that is required by the mitochondria. So அதுக்காக தான் உங்களுக்கு இந்த central dogma part வந்து உங்களுக்கு நான் explain பண்ணிருக்கேன். So this part which tells you about the central dogma. Okay, நீங்க ஒருத்தர் சொல்லிருக்கீங்க PCR வந்து இதுல வராது. PCRன்றது polymerase chain reaction. PCR எங்க எதுக்கு யூஸ் பண்ணுவாங்கன்னு கேட்டா amplification of DNA க்கு தான் யூஸ் பண்ணுவாங்க. எதுக்காக amplification of DNA? Amplification of DNA னா copies எடுக்கணும். For example, if you want to take a copies of a sheet, ஒரு xerox எடுக்கிறீங்க அப்படின்னா multiple copies எடுக்கிறீங்கன்னா அதுக்கு xerox பண்ற மாதிரி தான். ஒரு particular strand of DNA வந்து if you want to take the multiple copies, அந்த copies எடுக்கிற ப்ராசஸ்க்கு பேரு தான் polymerase chain reaction. PCR is a polymerase chain reaction, that is the amplification of DNA, where that particular, for example, MJ's Michael Jackson DNA, Mother Teresa's DNA, ஏதோ particular rare species of the DNA வந்து நீங்க நிறைய copies எடுத்து நீங்க research purpose யூஸ் பண்ணணும் அப்படின்னா you can go with the procedure called as polymerase chain reaction, where you can use an enzyme called as stack polymerase or enzyme which என்ன பண்ணுவீங்கன்னு கேட்டீங்கன்னா multiple copies எடுப்பீங்க. So that is and one more PCR ஓட advantage என்னன்னு கேட்டீங்கன்னா there you can also use it for where diagnostic procedure. Even nowadays you could see like corona la வந்து பாத்தீங்கன்னா how the diagnosing the coronavirus PCR polymerase chain reaction வச்சு இந்த blotting technique வச்சு they can identify. So PCR, western blotting, இந்த ELISA இதெல்லாம் techniques the molecular diagnostic perspective ல diagnosis கூட இந்த PCR technique வந்து யூஸ் பண்றாங்க. சரிங்களா? So don't get confused with the PCR in central dogma. PCR க்கு central dogma க்கு வந்து relation இல்லன்னு சொல்ல முடியாது. But coming to this part, there is no connectivity of PCR. Reverse transcriptase is the enzyme which is responsible for conversion of RNA to DNA. This process called as reverse transcription. So what is the use of protein produced by the central dogma? So எல்லாத்துக்குமே வந்து உங்களுக்கு protein தான் தேவை. உங்களோட பாடி மூவ்மெண்ட்ல இருந்து உங்களோட ஒவ்வொரு என்சைம்ஸ் பாடியில ப்ரொடியூஸ் பண்ற எல்லா என்சைம்ஸுமே என்னன்னா protein, because all enzymes are protein, but all proteins are not enzymes. உங்க பாடிக்கு தேவை எல்லா மெட்டபாலிக் ஃபங்க்ஷன்ஸ் A to Z எல்லாத்துக்குமே உங்களுக்கு enzymes and proteins தேவைப்படும். இந்த central dogma process நடக்கல அப்படின்னாக்கா proteins form ஆகாது. So proteins form ஆகல அப்படின்னாக்கா so you will not be like the cell cannot go for multiplication. Enzymes எல்லாத்துக்குமே enzymes தேவைப்படுது. Like right from digestion ல இருந்து உங்க neural system வந்து எல்லாத்துக்குமே you need an enzyme. So if proteins are not getting produced, then the person like the metabolic reactions and the cells which cannot go for a multiplication or the division as well. For that it requires a process called as central dogma அப்படின்னா replication, transcription, translation இது எல்லாமே வந்து நடக்கிறது compulsory. So protein synthesis compulsory நடக்கும். Okay, fine. So we done with this part, that is mitochondria. Next we move on to the next part. So the next part will start the cellular organelle is plastids. Coming to the plastid, so mitochondria முடிச்சிட்டோம். Next we'll start about the plastids. So plastids வந்து காமனா எதுல இருக்கும்? Animal cells ல இருக்குமா? Plant cells ல இருக்குமா? So which one controls the gene? It's a DNA or a protein அப்படின்னு கேட்டிருக்கீங்க. So controlling regulatory factors, regulation வரும்பொழுது of course it's actually like protein, the regulatory proteins சொல்லுவோம், regulate பண்ணும் proteins, but the one which controls the whole process actually the DNA, DNA which consists of a gene. சரிங்களா? ஆனா அதை இன்ஃப்ளூயன்ஸ் பண்ணி ரெகுலேட் பண்றது தான் யாருன்னு கேட்டீங்கன்னா proteins. Because proteins again இன்னொரு விஷயம், ப்ரோட்டீன்னா ஒரு ப்ரோட்டீன் கிடையாது, enzymes அப்படின்னு பாத்தீங்கன்னா ப்ரோமோட்டர் நீங்க எடுத்தீங்க லேக் கோப்ரான்ல படிக்கும்போது படிச்சிருப்பீங்க, டிரான்ஸ்அசிட்லேஸ், இந்த பீட்டா கேலக்டோசிடேஸ் இதெல்லாம் என்னன்னு கேட்டீங்கன்னா என்சைம்ஸ், அது புரோட்டீன் தான். சோ என்சைம்ஸ் என்ன பண்ணணும்னு கேட்டீங்கன்னா ரெகுலேட் பண்ணுமே தவிர ஃபுல் கண்ட்ரோல் யார் வச்சிருப்பான்னு கேட்டா ஜீன்ஸ் எங்க இருக்கும்? டிஎன்ஏல இருக்கும். சோ அப்படி அந்த ஆங்கிள் நீங்க பார்க்கும்பொழுது the DNA one which controls அதை ரெகுலேட் பண்ணி ஆன் அண்ட் ஆஃப் மெக்கானிசம் கொண்டு போகிறது யாருன்னு கேட்டா அது enzymes or the protein. Okay, so how many days it will take to identify the virus by using PCR technique? So PCR symptoms தெரியறதுக்கு தான் உங்களுக்கு வந்து 14 days ஆகும்னு சொல்றாங்க. சரிங்களா? பட் 14 days ஆன பிறகு PCR technique வந்து இட்ஸ் நாட் a very lengthy procedure. இது வந்து நீங்க கல்ச்சர் பண்ணி பிளேட்டிங் பண்ணி அந்த மாதிரி பண்ண வேண்டிய ப்ராசஸ் கிடையாது. இட்ஸ் a kind of blotting technique மாதிரி தான் பண்றாங்க. நீங்க படிச்சிருப்பீங்க southern blotting, western blotting, northern blotting. இந்த blotting technique மூலமா தான் PCR பண்றாங்க. Just within a like 1.5 days or 2 days கூட உங்களுக்கு PCR results வந்துரும். ஆனா சிம்டம்ஸ் வரதுக்கு தான் patient they have to wait for 14 days வரைக்கும் patients வெயிட் பண்ணணும். பட் PCR results வரதுக்கு வந்து PCR results வந்து 1.5 days 2 hours குள்ளே உங்களுக்கு PCR results கிடைச்சிடும். இது எதை டெக்னிக்கை பேஸ் பண்ணி பண்றான்னு கேட்டா blotting technique. நீங்க blotting படிச்சிருப்பீங்க. What is the blotting technique? There are 3 types of blotting இருக்கு. Northern blotting, southern blotting and western blotting. So இதுக்கு என்ன டிஃபரன்ஸ் இருக்கு? Northern blotting is for எதுக்கு பண்ணுவாங்க? Northern blotting, southern blotting, western blotting வந்து protein identify பண்றதுக்கு. Northern blotting வந்து use for the RNA, southern blotting is for the DNA. So அதனால ஈஸ்டர்ன் blotting இருக்கான்னு கேட்க கூடாது. ஈஸ்டர்ன் blotting கிடையாது. So we have only 3 types of blotting techniques. ஒரு மூணு விஷயம் தான். So DNA identify பண்ணனுமா, RNA identify பண்ணனுமா, protein identify பண்ணனுமா. DNA identify பண்ணனும்னா need to go for a southern blotting. நீங்க RNA identify பண்ணனும்னா you go for northern blotting. அதே நீங்க proteins identify பண்ணனும் அப்படினு கேட்டீங்கன்னா you need to go for a western blotting technique. The PCR அந்த technique கூட என்னன்னு கேட்டீங்கன்னா it's a kind of like the blotting techniques, the manipulated blotting techniques only they are doing. So PCR it won't take much time. So within like 1.5 like 20, I mean like 36 to 48 hours குள்ள உங்களுக்கு ரிசல்ட் கிடைச்சிடும். Good. So we go with the plastids. So ஏன்னா பிசிஆருக்கு வந்து இம்பார்ட்டன்ஸ் இருக்கு. ஸோ எதுக்கு நான் பிசிஆர் பத்தி இந்த இடத்துல நான் சொல்லியிருக்கேன்னு கேட்டீங்கன்னா பிகாஸ் டயக்னாசிஸ்க்கு வந்து பிசிஆர் யூஸ் பண்றதுனால யூ மே கெட் அ கொஸ்டின் ஃப்ரம் த பிசிஆர் சைட். பிசிஆரோட ஃபங்க்ஷன் என்ன, வாட் இஸ் அ பிரின்சிபல் பிஹைண்ட் த பிசிஆர். ஸோ இந்த இடத்துல இருந்து யூ கேன் கெட் அ கொஸ்டின். ஸோ டெஸ்ட் இஸ் பேஸ்ட் ஆன் பிளாட்டிங் ஆர் பை பிசிஆர். பிசிஆர் தான் பாலிமரேஸ் செயின் ரியாக்ஷன். பிசிஆரோட ஜஸ்ட் ஒரு மேனிபுலேட்டட் டெக்னிக் சம்டைம்ஸ் இந்த மற்ற சில வைரல் குரூப்ஸ்ல நீங்க போறதெல்லாம் பாத்தீங்கன்னா யூ ஹேவ் பிளாட்டிங் டெக்னிக். ஃபார் எக்ஸாம்பிள் யூ டேக் ஹெச்ஐவி இந்த மாதிரி கேசஸ் எல்லாம் பாத்தீங்கன்னா பிளாட்டிங் டெக்னிக்கை வச்சு என்ன பண்ணுவாங்கன்னா கன்ஃபர்மேஷன் பண்ணுவாங்க. எல்லாம் அட்வான்ஸ்ட் கம்ப்ளீட்டட் டெஸ்ட்டுக்காக தான் நீங்க பிளாட்டிங் டெக்னிக் பண்ண முடியும். கன்ஃபர்மேட்டரி டெஸ்ட்டுக்கெல்லாம் உங்களுக்கு பிசிஆர் போகுது. ஏன்னா 3 டெஸ்ட் இருக்கு எப்பயுமே. எதுவுமே ப்ரிலிமினரி ஒன்று இருக்கு, கன்ஃபர்மேட்டரி டெஸ்ட் இருக்கும், கம்ப்ளீட்டட் டெஸ்ட் இருக்கும். ப்ரிலிமினரி வந்து இந்த ரேபிட் கிட் மாதிரி சொல்றாங்க இல்லையா, இந்த ரேபிட் கிட் மாதிரி வரதெல்லாம் வந்து என்னன்னு கேட்டீங்கன்னா ப்ரிலிமினரியா பண்ற டெஸ்ட். கன்ஃபர்மேட்டரி டெஸ்ட் என்ன எலைசா இந்த பிசிஆர் எல்லாம் கன்ஃபார்ம் பண்ற டெஸ்ட். கம்ப்ளீட் பண்ணி ரீசர்ச் இன்னும் அட்வான்ஸ் டெக்னிக்ஸ் யூஸ் பண்ணி ரீசர்ச் டெவலப் பண்றதுக்காக தான் நீங்க பிளாட்டிங் டெக்னிக்ஸ் எல்லாம் யூஸ் பண்ணுவாங்க. ஸோ அதனால பிளாட்டிங் வந்து பிகாஸ் எக்ஸ்பென்சிவ் ப்ராசஸ். ஒவ்வொருத்தருக்கும் நீங்க பிளாட்டிங் பண்ணி பார்த்தீங்கன்னா இட்ஸ் வெரி எக்ஸ்பென்சிவ். ஒவ்வொரு பேஷண்ட்ஸ்க்கும் நீங்க பிளாட்டிங் பண்ணி பார்க்க முடியாது. இட்ஸ் டைம் கன்சியூமிங் ப்ராசஸ் அண்ட் ஆல்சோ இட்ஸ் எக்ஸ்பென்சிவ் ப்ராசஸ். அதனால பிளாட்டிங் வந்து மோஸ்ட்லி அட்வான்ஸ் கேசஸ்ல மட்டும் தான் பண்ணுவாங்க. கிளியர்? ஸோ பிஆர் இஸ் டன் யூசிங் blotting. So PCR is done using blotting. So it's a kind of completed. Blotting is a completed. Other moon type இருக்குன்னு சொல்றேன். So don't get confused with the PCR and the blotting. PCR is a separate test and blotting is a continuation of the PCR. Further உங்களுக்கு வந்து complete பண்ணனும் அப்படினாக்கா you want to identify certain research purpose use பண்ணனும், you can go for a blotting technique. Okay, fine. Now we'll start with the plastids. Coming to the plastids, this usually you could see in the plants. As far as the plastids are concerned, chloroplast. There are 3 types of plastid. Chloroplast, we have chromoplast. Chromoplast. And the third type of plastids we call it as leucoplast. So this part you can get a question. Plastids la the chloroplast la irundhu questions varum. So enna type of questions varuna the types la irundhu vandu questions varum. So coming to the chloroplast. Chloroplast which produce a pigment. What is the color of the pigment? The green color pigment. It's produced by this particular cells of the plastids that we call it as chloroplast, which is responsible for aligning a green color. The plants from the green color la irukana. This is because of what? This is because of this chloroplast, which ultimately will give green colors to the plants. So chloroplast which produce a green color pigment. Coming to the chromoplast. Chromoplast can be classified into phagoplast and rhodoplast. This can be classified into phagoplast and rhodoplast. So what is the difference? This can also produce a pigment. Chromo pigment produce panno. Chloroplast can also produce the pigment. Okay. So coming to the phagoplast and the rhodoplast. So phagoplast which produce the brown pigment, and rhodoplast which produce the red color pigment. Where you could see this brown color pigment. What is the name of the brown color pigment? It's a fucoxanthin. Fucoxanthin is actually the name of the pigment, the brown pigment. And coming to the rhodoplast, we have phycoerythrin. Example for the red pigment is phycoerythrin. Can you give some example where you could see? Of course, plants la irukkalam, but in the pigments எப்படி இருக்கும் பார்க்கிறதுக்கு brown color ஆ இருக்கும். So எங்க பார்க்கலாம் common the fucoxanthin and the phycoerythrin. Can you give an example where you could see more amount of this particular pigments, the brown pigments and red pigments are getting, I mean the fucoxanthin or phycoerythrin is getting produced. Algal species, நீங்க படிச்சிருப்பீங்க brown algae படிச்சிருப்பீங்க, the red algae படிச்சிருப்பீங்க. நீங்க red sea சொல்றீங்களா, the color of the red sea வந்து ஏன் அந்த ரெட்டாவே இருக்குன்னு கேக்குறீங்க. Because of this particular algae species, the red algae, this produces particular pigment called as phycoerythrin, and this phycoerythrin which keeps on producing the pigment, and then the color of the sea itself will become a red. This is because of the algal species like red algae, and this because the brown algae. Most of the algal species produce this type of pigment. Coming to the leucoplast classified. So coming to the leucoplast, these are all colorless. So this won't be having any sort of color. In the plastids, the leucoplast la vara in the plastids la enna pannuna, this will not actually produce what is it called, any colors, but The names that you could see is elaioplast, amyloplast, elaioplast, amyloplast, and there is one more leucoplast along with elaioplast, amyloplast. We have alleroplast. These are the three types of leucoplast. Elaioplast is for oil. This is for oil, and this is for starch, and alleroplast is for the protein. In the area you can get a question. So elaioplast வந்து எது ஸ்டோர் பண்ணி வைக்கும்னு கேட்டிங்கன்னா oil ஸ்டோர் பண்ணி வைக்கும். Amyloplast which store starch, and alleroplast which stores the protein. So this area, the plastids, you can get one possible question. Dinoflagellates ன்றது வந்து it's a kind of algae, and algae ல வந்து ஒரே ஒரு particular type of algae மட்டும் ரொம்ப பவர்ஃபுல்லான டாக்ஸின்ஸ் ப்ரொடியூஸ் பண்ணும். சரிங்களா? அது எந்த குரூப்பை சேர்ந்ததுன்னு கேட்டீங்கன்னா டைனோஃபிளாஜெல்லேட் அப்படின்னு சொல்லுவாங்க. திஸ் ப்ரொடியூஸ் லைக் ஃபைகோடாக்சின். மோஸ்ட்லி வந்து ஆல்கே வந்து மோஸ்ட்லி டாக்ஸிக்கா இருக்காது. ஆனா டைனோஃபிளாஜெலேட்ஸ்ல வர இந்த பர்டிகுலர் ஆல்கல் ஸ்பீஷீஸ் அக்வாட்டிக் இன் நேச்சர். இதுல வந்து ஸ்கூபா டைவிங் இந்த மாதிரி லைக் இதுல போறவங்க எல்லாம் வந்து மரைன் ஸ்டடிஸ் பண்றவங்க they should be very careful with this dinoflagellate, because this dinoflagellate is aquatic. It's a weak kind of thing, this algae which can produce the seed. Okay, so that is like the weak kind of thing which produce a toxin. This is called as phycotoxin, dinoflagellate. In the aspect la dinoflagellates மட்டும் கொஞ்சம் toxic தான். So chloroplast, chromoplast, leucoplast. So the three types of leucoplast: elaioplast, amyloplast, and alleroplast. Elaioplast is for storing oil, amyloplast is for the starch, and alleroplast is for the protein. So in the area la you can get a question. இதுல ஏதாவது ஒரு டைப் கொடுத்துருவாங்க. So elaioplast is for the storing of ஆப்ஷன்ஸ்ல வரும். அப்போ elaioplastனா ஆயில் உங்களுக்கு கன்ஃபார்மா தெரியும். So amyloplast வந்து ஸ்டார்ச் தெரியணும். alleroplast வந்து protein. Or sometimes they could ask in the match மாதிரி கேட்பாங்க. column A, column B மாதிரி questions கேட்டு அதுல வந்து நம்ம மேட்ச் பண்ணிப்போம். So இந்த ஏரியால especially leucoplast இந்த ஏரியால வந்து you can get a question. So இதுல கொஞ்சம் பார்த்துக்கங்க. So third type of leucoplast வந்து alleroplast, and this alleroplast responsible for the storage of protein. Clear guys? So we're done with the plastids. Now full structure up சொல்லல, because plastids la in the area la you can get a question. Next we move on to the chloroplast. Coming to the chloroplast. As far as chloroplast is concerned, we start with the chloroplast. So mitochondria பார்த்தோம், then we discussed about plastid, and then comes your chloroplast. Chloroplast வந்து which usually the thickness அப்படின்னு பாத்தீங்கன்னா 90 to 100 angstrom. The thickness of chloroplast is 90 to 100 angstrom thickness. So it performs important functions in the protein synthesis. This consists of outer membrane. This part is outer membrane. See, this outer membrane, and we have inner membrane. And inside, when you go inside, you could see the matrix part, and the matrix, this is a matrix, this region is a matrix region. This matrix which consists of stroma. And even go deeper inside, you could see like there is a granum part, and there you could see the coin, like it gets stacked on each other. Coins வந்து மேல அடுக்கி வச்சிருக்க மாதிரி ஒரு ஸ்ட்ரக்சர்ஸ் இருக்கும். So this structures we call it as a granum. It's a granum. கிரானம் குள்ள இருக்கிற இண்டிவிஜுவல் ஒவ்வொரு யூனிட் இருக்கு பாருங்க, இந்த ஒவ்வொரு யூனிட்டும் வந்து we call it as a thylakoids. அப்படின்னா if fusion of a complete collection of the thylakoids, that we call it as a granum ன்றது complete structure. அந்த கிரானம்ல இருக்கிற ஒவ்வொரு தனித்தனி யூனிட் இருக்குல்ல, அந்த யூனிட்டை தான் நம்ம என்ன சொல்லுவோம் கேட்டா thylakoids அப்படின்னு சொல்லுவோம். சோ இந்த ஃபுல் ஸ்ட்ரக்சர் பேரு கிரானம், கம்ப்ளீட் ஸ்ட்ரக்சர். இது ஒரு கிரானா, இது ஒரு கிரானா, இது ஒரு கிரானா, இது ஒரு கிரானா, கிரானா கம்ப்ளீட் கிரானா. டுகெதர் we call it as a grana. But கிரானக்குள்ள இருக்கிற ஒரு தனித்தனி ஸ்ட்ரக்சர்ஸ் இருக்கு பாருங்க, இது ஒரு thylakoid, this is a thylakoid. So you have a successive like piling up of like thylakoids which present one above the other. So in the thylakoids only which is responsible for the protein, which is responsible for photosynthesis. Another main thing that you should understand between the grana and the stroma, two types of reaction takes place. Photosynthesis படிச்சிருப்பீங்க. What are the two types of reaction? Light reaction and dark reaction. இது எப்படி கனெக்ட் ஆயிருக்கும் கேட்டீங்கன்னா, this grana is connected by lamellae. The stroma lamellae. So connective ஒரு கிரானாவும் இன்னொரு கிரானாவும் எப்படி கனெக்ட் ஆயிருக்கும் கேட்டீங்கன்னா by means of a structure called as lamellae. Okay, lamellae. Fine. So you asked me like, sir, why the sizes differ between the chloroplast and the ribosomes. The size, because the size vary in the chloroplast, the majority of the functions which takes place here is the photosynthesis. Because when you study the respiration chapter படிக்கும்போது chloroplastல வந்து அதிகமான வந்து photosynthesis process நடக்கும். Two types of reaction நடக்கும். What are the two types of reaction? Light reaction and the dark reaction. இந்த process நடக்கும்போது the ribosomes on the size வந்து it should be like in a very less. So another chloroplast comparatively ribosomes the size வந்து will be very less one. So the light reaction and the dark reaction. The light reaction and dark reaction. The ரெண்டு reaction வந்து chloroplast தான் நடக்கும். Now can you tell me like which part is responsible for the light reaction and which part is responsible for the dark reaction? So granum is responsible for granumல என்ன reaction நடக்கும்? ஒன்னு வந்து granumல நடக்கும். The other one will take in stroma. So granum is responsible for the light reaction. Light reaction takes place in the granum, and in a stroma, dark reaction takes place. So both light reaction as well as dark reaction, both is actually the cycles successive reactions takes place. It's for photosynthesis. So light reaction, the presence of light, which means the daytime, the presence of sunlight. You could see that could occur in a granum. Granum எங்க thylakoidsல வந்து நடக்கிற process light reaction. அதே வந்து நீங்க வந்து இந்த பத்தி and we are talking only about the cell organelles. The detailed structure and functions நம்ம எப்போ படிப்போம்னா when we talk about the plant physiology ல வந்து நம்ம thylakoids இந்த granum ஓட complete structure, the bundle sheath cell, C3 plants, C4 plants, CIM plants, இந்த மாதிரி ஒரு categorize பண்ணி we're going to discuss in detail when we talk about the plant physiology. So dark reaction takes place in the so இதுல இருந்து questions வரும். Dark reaction, which part of the chloroplast responsible for the dark reaction? அப்போ options கொடுத்திருப்பான். அவன் thylakoid கொடுத்திருப்பான், grana கொடுத்திருப்பான். இந்த மாதிரி options போகும்போது you need to select stroma which is responsible for the dark reaction. Light reaction அப்படிங்கும்போது grana which is responsible. So light and dark reaction is a part of photosynthesis. These reactions takes place. Okay. So can someone tell me like how many thylakoids? So ஒரு particular cell எடுத்துக்கிறீங்க அப்படின்னா ஒரு chloroplast எடுத்துக்கிறீங்க. அந்த chloroplastல வந்து maximum எவ்வளவு chloroplast இருக்கும்? So actually this is not a definite number வந்து சொல்ல முடியாது thylakoidsல வந்து because it keeps varying. வேணா C3 plants ஒரு மாதிரி இருக்கும், C4 plants ஒரு மாதிரி இருக்கும், and CAM pathway. Okay, so ஒவ்வொரு செல்ஸ்க்கும் the one type of plants to the other type of sugar cane ஒரு மாதிரி இருக்கும், cotton ஒரு மாதிரி இருக்கும். So ஒவ்வொரு plant to plants like this thylakoids number which keeps varying. So the thylakoids number which cannot give a definite number for particular இவ்வளவு தான் இருக்கும் அப்படின்னு சொல்லுவாங்க. This thylakoids which keeps varying from one plant to the other plant. Fine. So this is about the chloroplast structure. And we move to the next part. And who discovered and who coined this chloroplast? This was coined by Schimper, a person named Schimper. Schimper is a person who coined the chloroplast பேரை கொடுத்த ஒரு பேர் என்னன்னு கேட்டீங்கன்னா Schimper in the plants actually. So நீங்க கேட்டீங்க C3 C4 plants என்ன அப்படின்னு கேட்டீங்கன்னா you could see the presence of chloroplast in a bundle sheath cell. Bundle sheath cell வந்து chloroplast வந்து இருக்கிறதையும் இல்லாததையும் பேஸ் பண்ணி தான் will differentiate C3 and C4 plants. So ஒரு bundle sheath எடுத்துக்கிட்டீங்கன்னா அதுக்குள்ள வந்து chloroplast வந்து C3 இருக்கும், அதே வந்து C4 ல வந்து இருக்காது. So based on the presence of the chloroplast in the bundle sheath cell, this C3 and C4 plants can be categorized. And other difference என்னன்னு கேட்டீங்கன்னா C3 க்கும் C4 க்கும் the first stable compound which forms is a 3-carbon compound in case of C3. C4 plants la the first stable compound which forms during the photosynthesis, it's a 4-carbon compound. Adanada the C3 no C4 no solvom. But principle wise pathina C3 la we could not see the presence of the chloroplast in bundle sheath, but C4 la you could see the presence of chloroplast in the bundle sheath cell. Based on this, you can differentiate. C3 pathina cotton plants you can take, cotton beans alla edukalam. C4 la for example you take sugarcane. These are which comes under the C4 plants. So we need not worry about the C3 and C4 light reaction and dark reaction. The principle pathi we need not worry at this point of time. Number we're not discussing about the reactions. Inga number reactions pathi paaka poradhu. Number structure pathi dhaan paakarom. In detail number enga padipom kettinga plant physiology especially photosynthesis padikumbodhu na idha pathi ungalku detail ah solli tharan. Okay, I hope I answered for your question. C3 and C4 chloroplast irukradhu illadha vechi differentiate பண்ணுவோம். இன்னொரு difference C3 க்கும் C4 க்கு என்ன difference na stable compound. The first stable compound வந்து 3-carbon compound இருந்ததுன்னா அது C3. The first stable compound which formed during photosynthesis வந்து 4-carbon compound இருந்துச்சுன்னா that is called as C4 plants. And second difference is chloroplast presence and chloroplast absence. Either based பண்ணி we differentiate C3 and C4 plant. Between the chloroplast and the mitochondria. Of course, a cell organelle அப்படி இருக்குன்னு கேட்டீங்கன்னா mitochondria வந்து which is giving actually power ATP. இந்த process நடக்கும்போது plastids, for example, this chloroplast வந்து photosynthesis process நடக்கும்போது definitely it requires ATP. அப்ப ATP யார் produce பண்ணுவா? Mitochondria. So definitely there will be signal mechanism which occurs between your chloroplast and the mitochondria. Similarities may be what in the sense like structure wise the matrix in the region which is similarities பார்க்கலாம், but function wise there won't be much of similarities between your mitochondria. ஆனா dependency compulsory இருக்கும். chloroplast வந்து நீங்க photosynthesis நடக்கும்போது definitely it requires ATP. That point of time, of course, there is a what is it called, connectivity between your mitochondria and of course with the chloroplast. Okay, so we're done with the chloroplast. Next we move on to the ribosomes. Next part we'll discuss about the ribosomes. We not much discuss about the ribosomes because you know already about the ribosome. The ribosomes are the smallest cell organelle that I already told you. As far as the ribosomes are concerned, usually it is 150 to 200 angstrom diameter. 150 to 200 angstrom in diameter. This is actually in diameters. This is ribosomes, and this doesn't have any membrane. And who first identified this ribosome? It's Pellet. So sometimes pellet particles. The name of the person who first identified is Pellet in the year 1955. 1955, he first identified the ribosomes, and for this ribosome formation, it requires magnesium ions. Ribosome formation important ions in it requires magnesium ions. In case magnesium ions அதிகமா இல்லாத பட்சத்துல ribosome formation வந்து செல்லுக்குள்ள ப்ராப்பரா இருக்காது. அப்போ ரைபோசோம்ஸ் கரெக்டா இல்ல அப்படின்னு கேட்டீங்கன்னா எது நடக்கிறது கம்மியாகும்? புரோட்டீன் சிந்தசிஸ். ஏன்னா புரோட்டீன் சிந்தசிஸ் கம்பல்சரி எது வேணும்? ரைபோசோம்ஸ் வேணும். So as for ribosomes are concerned, ribosomes are classified into two types, 70S or 80S or we can classify. 70S you could see in case of prokaryotic cell, 80S you could see in case of eukaryotic cell. Okay, eukaryotic cell. See, as for the dates are concerned, நான் கொடுக்கிற dates வந்து எப்படின்னு கேட்டீங்கன்னா நீங்க 1953 ன்னு சொல்றீங்க. 1953 yes, அவங்க initial point of time study conduct பண்ணது வந்து 1953. பட் அது அப்ரூவல் நான் எப்பவுமே நாங்க கொடுக்குற டேட்ஸ் எப்படின்னு கேட்டீங்கன்னா when the research has been அப்ரூவ் பண்ண இயரை நான் கொடுத்துருக்கேன். ஸோ இன் கேஸ் நீங்க எதை சார் ஃபாலோ பண்ணணும் அப்படின்னு கேட்டீங்கன்னா you follow with the textbook the 1953. பட் இங்க 1955 கொடுத்துருக்க ரீசன் வந்து his work has been approved only in the year 1955. But our period of the study begin பண்ணது 1953. So you follow the textbook itself, no issues. 1953 is நீங்க போடுங்க. So 70 years and 80 years. So 70 years for prokaryotes. What does the S stands for? S இருக்குல்லையா, S எதை denote பண்ணுது? In ribosomes, what does the S stands for? There's a name, right? The meaning for the S. So what does the S stands for? Coming to this ribosome. This ribosomes which consist of special protein, these proteins are called as riboforins. This called it as riboforins. The special types of proteins where the ribosomes are made up of, it is actually riboforin. So S stands for Swedberg unit. Based on the sedimentation coefficient only, we are categorizing the 70S and 80S. The 70S 80S கூட நீங்க பார்க்கும்போது எப்படின்னு கேட்டீங்கன்னா ஒன்னு வந்து smaller unit இருக்கும், இன்னொன்னு வந்து larger unit இருக்கும். So if you could see the ribosome structure, for example, this is your smaller unit and one will be your larger unit. This is a smaller unit and this one is the larger unit. So like this you can categorize again, like how this can be like differentiated as smaller unit and the large by means of sedimentation coefficient, right? Again you need to understand ribosomes and the specialized structures சொல்றோமே அது வந்து எதை வச்சு it's made up of what? It's made up of the rRNA. It's made up of the rRNA. நீங்க 3 types of rRNA படிப்பீங்க. என்னென்ன type of RNA are three main important the types of RNA? We have messenger RNA, ribosomal RNA and we have a transfer RNA. Sometimes like heterogeneous RNA இந்த மாதிரிலாம் கூட படிப்பீங்க. But translation process நடக்கிறதுக்கு 3 important RNAs required: mRNA, rRNA and tRNA. But the ribosomes in the structure வந்து made, how it is actually made, this is by means of accumulation of the ribosomal RNA. So difference between the chloroplast ribosomes and cytoplasmic ribosomes, as already told you, like chloroplast is the way the photosynthesis process which takes place, that is a light reaction and a dark reaction, right? So this ribosome should not be like interface for this particular process. So that be definitely the difference between the size of the ribosomes between your the cytoplasmic ribosomes and the chloroplast ribosomes. Adanaladha difference compulsory irukkum. Okay. Fine. So riboforins that we discussed are the small unit, and we discussed about the large unit. So we discussed about the the ribosomes. Bacterial cells la pathina, how many ribosomes that you could see in a bacterial cell? அதே வந்து ஒரு eukaryotic cell எடுத்துட்டீங்கன்னா எந்த ரேஞ்சுக்குள்ள வந்து உங்களுக்கு வந்து ribosomes இருக்கும். So normally you take the bacterial cell, normal bacterial cell which consists of usually it consists of 10,000 the ribosomes that you could see. But in case of eukaryotic cell, there's a range between 1 million to 10 million ribosomes which present in the eukaryotic cell. Definitely prokaryotes and eukaryotes number of ribosomes vary agum. Idhula for example 10,000 varkum da irukum bacterial. Adhe vandu eukaryotes eduthinga range which goes between 1 million to 10 million varkum you could see the presence of ribosome. Okay, fine. Yes, of course, the food can be digested in the vacuole. That is also the important functions of the vacuole. Very shortly, we discuss about the vacuole also. Okay. Next, we move on to the endoplasmic reticulum. So coming to the next part, endoplasmic reticulum, we simply call it as ER. ER stands for endoplasmic reticulum. Endoplasmic reticulum. This endoplasmic reticulum is the invaginations of the nuclear envelope. Nuclear envelope the nuclear envelope invaginations. So you could see the invaginations, the foldings, the structure. This is what actually talking about, the endoplasmic reticulum. Some endoplasmic reticulum will be rough in nature. Slightly rough smooth. So what does it denote? The rough endoplasmic reticulum. So this is called as rough endoplasmic reticulum. And there is some part, the other part of the endoplasmic reticulum is considered to be SCR, that is smooth endoplasmic reticulum. So based on what this can be categorized, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough is actually, you could see the presence of ribosomes on its membrane. So we could see the presence of ribosomes on its structure. This denotes rough endoplasmic reticulum. And here in this case, you could not see the ribosomes on its surface. So it is called as smooth endoplasmic reticulum. And this was first reported, the endoplasmic reticulum was first reported by Porter and Cloud. Reported by a person named Porter and Cloud in the year 1945. And later part, actually, this was discovered completely by a person named Coleman in the year 1952. So 1952. As far as endoplasmic reticulum is concerned, this is the thinner one, and the size of this is 5 angstrom. It's very thin. The endoplasmic reticulum, it is 5 angstrom. It's a 5 angstrom. What is the composition of endoplasmic reticulum? This consists of 30 to 50% of lipids. 30 to 50% of lipids. 30% and 30 to 50% lipids, and the remaining part is usually we have got phospholipids. The remaining 70% which consists of the specific type, we call it as phospholipids. So 30% of normal lipids, remain the 70% more precisely we have a phospholipids. Clear with this one? And coming to the parts that you could see in endoplasmic reticulum, cisternae, tubules and vesicles. Cisternae, 3 parts irukku. Cisternae, tubules, and we have a vesicles. So together we call it as an endoplasmic reticulum. So endoplasmic reticulum, there are 3 parts where you could see cisternae, tubules, and vesicles. Coming to the cisternae, this is with the ribosome. You could see the ribosome is present. The ribosome is present here. Here there is no ribosome. The ribosome is not present in the tubular region. Coming to the vesicle region, it's ovoid and it's like isolated sacs. It's a like sac-like structure. Here also you not see the ribosomes. So 3 important regions: cisternae, tubules, and the vesicle parts. And the first part, the cisternae, you could see the presence of ribosome. In other areas like tubules and vesicles of endoplasmic reticulum, where you could not see the presence of the ribosomes. Coming to the functional area, where the smooth endoplasmic reticulum, rough endoplasmic reticulum, which plays a very important role in synthesis of protein. More specifically, in the rough endoplasmic reticulum is responsible for the synthesis of protein. Smooth endoplasmic reticulum is responsible for synthesis of lipids and steroids. This is responsible for lipids and steroids. SCR, what is the important main functions of SCR? SCR is responsible for synthesis of lipids and steroids. Coming to the rough endoplasmic reticulum, this plays an important role in synthesis of the protein. In this way, like you can differentiate RER and SCR. So we done with the smooth endoplasmic, and also SCR also, which plays a very important role in the detoxification of drugs. Detoxification of, this is also the additional function. For example, you take a drug, actually the remaining drugs like which is going at the wastage, which is going to a cell. So who is detoxifying the remaining like constituents of like surplus the drugs? Who is detoxifying? The side effects varama paakuradhu yarunu kettingna your smooth endoplasmic reticulum. The smooth endoplasmic reticulum is responsible for detoxification of the drugs, and also this is responsible for synthesis of steroid hormones, and also smooth endoplasmic reticulum, which acts like the storehouse of calcium ions. This also act like a storehouse of storehouse of calcium ions. These are the very specific function that you could see in this case. You ask the reason, sir, telling food is digested in a vacuole. That what about the stomach? It can be understand the concept. See, the food is getting digested in the vacuole and talking about like process called as I told you like bulk transport mechanism, pinocytosis. I told you exocytosis, endocytosis, that is phagocytosis and pinocytosis. It's happening in the cell. So don't connect with this process. I'm talking about the cell. I'm not talking about any system. So cell tissue and cell எல்லாம் சேர்ந்தது, மசில் என்ன டிஷ்யூ எல்லாம் சேர்ந்தது. ஸோ நான் சொல்றது ஒரு செல்லுக்குள்ள மட்டும் நடக்கிற ப்ராசஸ். செல்லுக்குள்ள இருக்கிற ஒரு வேக்வல் மட்டும் சில ஃபுட்டை வந்து டைஜஸ்ட் பண்ற கேப்பபிலிட்டி இருக்கு. நீங்க சொல்றது வந்து எதுன்னு கேட்டீங்கன்னா சிஸ்டம் சொல்றீங்க, டைஜஸ்டிவ் சிஸ்டம். ஒரு சிஸ்டமே நீங்க எடுக்கும் பொழுது அப்படின்னு கேட்டீங்கன்னா the digestion definitely takes, அது கூட ஸ்டொமக்ல மட்டும் நடக்காது. ஸ்டொமக் இருக்கு, டியோடினம் இருக்கு, then absorption இருக்கு, assimilation, different mechanism நடக்க வேண்டியது இருக்கு. The digestive system is completely different, where lots and lots of cells and enzymes or proteins are involved in this particular process. I hope I answered for this question. But உங்க வேக்வல்ஸ் நீங்க எடுக்கிறீங்கன்னா வேக்வல்ஸ் வந்து ஒரு பர்டிகுலர் செல்லுக்குள்ள இருக்க ஒரு ஸ்ட்ரக்சர் பத்தி மட்டும் பாக்குறோம். அப்போ அந்த செல்ல வந்து வேண்டியதை எடுத்துக்கிறதோ இல்ல வேண்டாத வெளியில அனுப்புறதுக்கும் யூஸ்ஃபுல்லா சம்டைம்ஸ் வேக்வல்ஸை யூஸ் பண்ணிக்கும். அந்த இடத்துல நீங்க வேக்வலை பத்தி ரெப்ரசென்ட் பண்ண, அது வந்து phagocytosis இருக்கலாம், செல் ஈட்டிங், பினோசைட்டோசிஸ் அல்ல, செல் ட்ரிங்கிங் மெக்கானிசம். This by means of this mechanism this process can occur, right? In case of like you're talking about the vacuole and also maintains buoyancy of the cell. அது ஒரு செல்ல பத்தி மட்டும் பேசுறதா இருந்தா you're talking about the vacuole. But ஒரு சிஸ்டமோட நீங்க கம்பேர் பண்ணும்போது you cannot compare with the vacuoles, because the system comprises about what? Comprises about the complex mechanisms like lots of enzymes and proteins and other type of like the complex structures also involved. For example, in stomach the argentine cells படிச்சிருக்கீங்க, oxyntic cells படிச்சிருக்கீங்க, chief cells படிச்சிருக்கீங்க. The varied group ஒரு stomachல எடுத்துட்டீங்கன்னா 6 types of cells are involved in this, which plays a very important role in the digestion process. One of the gastric secretion, one of the hydrochloric acid secretion, one of the precursor. இந்த மாதிரி நீங்க 6 type of cells படிச்சிருப்பீங்க. I hope you can recollect the things that we discussed in the digestive system. அந்த இடத்துல வந்து vacuolesல இருந்து ஒரு particular cell பத்தி நம்ம டிஸ்கஸ் பண்ணலாம், because it's a complete system. So I hope I clear with this part. So don't get confused with the stomach. நம்ம ஸ்டொமக்ல நடக்கிற டைஜஷனோ ஒரு செல்லுக்குள்ள டைஜஷன் வந்து, of course ஸ்டொமக்ன்றது வந்து செல்லால மேட் அப் ஆஃப் ஆயிருக்கிறது மட்டும்தான். ஆனா வேக்வலால மட்டும்தான் இந்த டைஜஷன் நடக்குதுன்றது it's not correct. This because happening by means of the enzymes and by means of various proteins and glands. இந்த நிறைய விஷயம் இருக்கு. Okay, I hope we already discussed about this in the digestive system. Okay, coming to back to this part, the smooth endoplasmic reticulum. So detoxification drugs, and also this act like the storehouse of the calcium ions. So we done the smooth endoplasmic, that rough endoplasmic reticulum. Then we move on to the next part. Next part is Golgi apparatus. Coming to the Golgi apparatus. As far as who discovered Golgi apparatus? So what's the name of the person who discovered Golgi apparatus? It's a membranous granule which is present inside the cell. It's a flattened sac-like structure. It's a flattened sac-like structure. This was first proposed by Camillo Golgi. He's a person first identified this Camillo Golgi in the year 1897. Again, I'm telling you, if you're not this comfortable with the year I'm mentioning here, if you could see there's some other like year that is mentioned textbook, please follow the NCERT itself, because if you, this year seems to be not matching, so you need not worry about this year. Okay, if there's some other year is given in the textbook, please follow your NCERT. Fine. But the common terms like Camilla Golgi, he first identified this Golgi apparatus in the year 1897, and he's got the Nobel Prize for this invention in the year 1906. He's a person who got the Nobel Prize for this invention in the year 1906. 1906. Coming to this part where the Golgi apparatus is a membranous organelle, I told you. As you could see, this structure, this Golgi apparatus, this also can be called as dictyosomes. This can be also called as sometimes dictyosomes and kudukranga appdin ketanga adu edha solranga appdin kettinga they're talking about the dictyosomes here. So they're talking about the dictyosomes in case of plants and fungi. Usually in case of plants and fungi, they precisely they use a word called as the dictyosomes. And here you could see the structure like, you could see the structures like this, in a very disc-like structures, one above the other. You could see as far as your memory phases, you could see, you would have noticed like the structures of the Golgi apparatus. Here are two sides. One is the convex side, other one is the concave side. So convex side is called as convex side, and the other one is the concave side. So the convex side. So the convex side is actually called as cis phase, and this one is called as trans phase. We have a cis phase and two phases that you could see, that is cis phase and the trans phase. And you could see also the production of the lysosomes. From the vesicular part, you could see the production of the lysosomes. These lysosomes, which consist of an enzyme called as lysozyme, the vesicle-like structure, right, which bulge out, and the terminal part, they start producing this vesicle-like structure. This vesicle is called as lysosome. This lysosome, which consists of enzyme called lysozyme. So there is a connectivity with the Golgi apparatus and the lysosome. Okay, so here the lysosome, which consists of a lysozyme enzyme. Coming to the cistern part. The cistern part only I told you, like, the convex and the concave. So cistern is actually the flat, the membranous structure. This part is only called as a cistern. This part is called as cistern, and this cistern consists of two regions. One is a convex part, other one is a concave part, right? And this convex is actually cis phase, and the concave, which is called as a trans phase, and the lysosome, which is also called as a suicidal bag. So can you someone tell me, like, why this lysosome is called as suicidal bag? ஏன் வந்து suicidal bag ன்னு சொல்றாங்க? எதை suicide எதை வந்து destroy பண்ணும்? எதுக்கு particular வந்து this lysosome that we call it as a suicidal bag? There's a particular reason for that. எதை actually eliminate பண்றதுக்காக, எதை it's going to destroy, so that unwanted things are destroy பண்றதுனால, இல்ல செல்லுக்கு important things are destroy பண்றதுனால, செல்லையவா இல்ல dead செல்லையா இல்ல aged செல்லையா, எந்த particular compounds இது destroy பண்றதுனால, the lysosomes that we call it as the suicidal bags of a cell. So it destroys itself. It can digest its own cell. So once the older cells, where the half-life days of the particular span of the life, the cell is getting completed, or the cells like which is not completing its cell cycle, right? Then, and most of the time it's being in the GO phase of the cell cycle, then particular time what happens, like the cell itself will be destroyed by means of the lysosome. So we call it as the suicidal bags of the cell. Maybe the aged normal cells, metabolically active agra cells destroy panichna, then we cannot survive. So ஒவ்வொரு டிஷ்யூஸ்க்கு வந்து மெட்டபாலிக் ஆக்டிவான செல்ஸ் தேவைப்படும். எல்லா செல்ஸும் டிஸ்ட்ராய் பண்ண முடியாது. ஸோ இட் யூஷுவலி டிஸ்ட்ராய்ஸ் த ஏஜ்டு செல்ஸ் ஆர் தி டேமேஜ் செல்ஸ். அந்த செல்லால ஆக்டிவாக பயாலஜிக்கல் ஆக்டிவாக இருக்க முடியாது. அந்த மாதிரி பட்சத்துல என்ன பண்ணும் அப்படின்னு கேட்டீங்கன்னா, the cells can be eliminated. This is happened by means of lysosomes. இது வேலையை பண்ணும். அதனாலதான் lysosomes என்ன பண்ணும் சொல்லுவோம் கேட்டா, it's called as suicidal bags of the cell. Okay, we're done with the Golgi apparatus. It produces a lysosome. And this also, like, very could see the lysosome. You could also see this lysosome in an acrosome. Acrosome enga paathirpeenga, you could see in a sperm. The sperm head la vandu we have the terminal part, right? The acrosome, the sperm head, which consists of the terminal part. This terminal part, which consists of again lysosome, because the lysosome, which plays a very important role in the penetration. So when it penetrates at the fertilization time, the sperms get penetrate with the ovum. How this process occurs is because of the enzyme which present at the tip, the head part of the sperm. Again, this is a lysosome. So that is the function again by the lysosome. And this lysosome, which consists of proteins and also the lipids. So proteins, lipids, which formed in what? Which forms in the endoplasmic reticulum. Endoplasmic reticulum form on the proteins, lipids வந்து எதோட combine ஆகும்னு கேட்டீங்கன்னா, carbohydrate ஓட combine ஆகும். So carbohydrate ஓட combine ஆகும்போது what happens, like protein protein plus the lipid which has been formed in the form of endoplasmic reticulum form actually. In the protein and the lipid which will be coming here, which will be coming and to the which combines with the carbohydrate. This combines with the carbohydrate and this gets converted. This gets converted to glycoprotein. Glycoprotein and glycolipid are marked. Golgi apparatus it will be converted. So proteins and lipids have been formed in the endoplasmic reticulum. Protein and lipid which has been formed in the endoplasmic reticulum, and this gets converted to carbohydrate. Carbohydrate மாறும்போது என்ன ஃபார்ம்ல ஆகும்னு கேட்டிங்கன்னா proteins will be converted to glycoprotein. Lipids will be converted to glycolipid. Now this glycoprotein and glycolipids which comes to the Golgi apparatus. இந்த இடத்துல வந்து of course there is a connectivity. எதுக்கும் connectivity இருக்குன்னு கேட்டிங்கன்னா endoplasmic reticulumக்கும் உங்க Golgi apparatusக்கும் connectivity இருக்கு. Okay, so no cell organelles which can function as an individual entity. புரியுது, நம்ம உடம்புல வந்து ஒரு எல்லா ஆர்கன்ஸும் ஒண்ணா சேர்ந்து வேலை செய்யற மாதிரி ஒரு செல்லுக்குள்ளேயுமே எல்லா ஆர்கனல்ஸும் வந்து which has to be coordinated. The same way, here is an example I could see. The endoplasmic reticulum which produces protein and lipid which is getting converted to carbohydrate and which is coming to the Golgi apparatus in the form of glycoproteins and glycolipid. Fine. Next, we move to the next part, lysosomes. The next part is lysosomes. And the vesicle-like structure which produced out of the Golgi apparatus, we call it as lysosome. So this, I already told you, this is called as suicidal bag. Lysosomes is otherwise called as suicidal bag of a cell. This is otherwise called as garbage disposal system. This can be also called as garbage. The common name that we'll give is a garbage disposal. Between garbage disposal system சொல்றாங்க, எதை சொல்றாங்க அப்படின்னு கேட்டீங்கன்னா they're talking only about the suicidal bags, that is lysosomes. Clear? And this consists of the lysosomes in the particular property this consists of hydrolytic enzymes. Lysosomes consist of an enzyme called hydrolytic. This consists of hydrolytic enzymes. And mostly what happens, this is not present in RBC. The lysosome it's absent. It's absent in RBC. It's also absent in a bacteria and also like in a prokaryotic cell, and also this you could not see in a plant cells. Plant cells you cannot see this type of the lysosomes. அதனால தான் வந்து பாக்டீரியால வந்து for example நீங்க tears உங்க சலைவல எல்லாம் பாத்தீங்கன்னா எது இருக்குன்னு கேட்டீங்கன்னா இந்த லைசோசைம் இருக்கும். இந்த லைசோசைம் இருக்கிறதுனால தான் என்ன ஆகுது அப்படின்னு கேட்டீங்கன்னா இட்ஸ் இஸ் a bactericidal. உங்க சலைவா வருதுல உங்களுக்கு கண்ணுல இருந்து வர கண்ணீர் இந்த டியர்ஸ்ல கூட என்ன இருக்குன்றாங்க, லைசோசைம் இருக்கு. அது பேசிக்கா என்ன ப்ராப்பர்ட்டி இருக்குன்னு கேட்டீங்கன்னா பாக்டீரிசைடல். அப்படின்னா ஒரு பாக்டீரியா வந்து சாகடிக்கிற தன்மை இருக்கு. அப்ப இதுக்கு என்ன அர்த்தம் கேட்டா பாக்டீரியாக்கு ஜெனரலாவே என்ன ப்ராப்பர்ட்டி கிடையாதுன்னு கேட்டா லைசோசோம் ப்ரொடியூஸ் பண்ற அந்த எபிலிட்டி வந்து கிடையாது. So we discussed already about lysosome, what it's called as suicidal bags in the previous part itself. We discussed because it used to eliminate all the dead cells and the aged cells which not performing a biologically active mechanisms. அப்படி cells destroy பண்றதுனால this we call it as suicidal bags. தேவையில்லாததை ரிமூவ் பண்றது கார்பேஜ், அதாவது இந்த சிஸ்டம் பேரு கார்பேஜ் டிஸ்போசல் சிஸ்டம். ஓகே, so we discussed about lysosomes and the structure. As the structure is concerned, we have two types of lysosomes. One is primary lysosome and we have a secondary lysosome. Primary lysosomes which formed from endoplasmic reticulum. This is in the form of formed by endoplasmic reticulum. The secondary lysosome which is formed by the fusion of phagosome and the lysosome. Which is formed by the fusion of phagosome and lysosome. நீங்க கேட்டீங்க WBC ல வந்து உங்களுக்கு வந்து இந்த லைசோசோம்ஸ் இருக்குமா? கண்டிப்பா இருக்கும். because WBCs are phagocytic cells. இந்த phagocytic cells cell eating mechanism, phagocytosis அப்படின்னு நம்ம சொல்றோம்ல, WBC ஏன் சொல்றோம்? cell eating mechanism. ஒரு பாக்டீரியா வருது, for example this is considered as a WBC. இது வந்து பாக்டீரியா. you take this is a bacterial cell. Okay, this bacteria has to be engulfed. This WBC, this is a WBC. This WBC which consists of lysosome. அப்ப என்ன பண்ணுவோம்னா this bacteria which comes inside, which engulfed inside, and this structure is called as phagosome. So what happens, the lysosome, this is the lysosome, the lysosome which fused with the bacteria, right? And this forms a phagosome. And then what happens, this lysosome and phagosome fuse together and form a structure called as phagolysosome. This is called as phagolysosome. இந்த phagolysosome என்ன ஆகும்னு கேட்டீங்கன்னா, the lysosome which present in the sac-like structure which acts on the bacteria, and eventually what will happen? The bacterial cell will be, the antigen will be destroyed, and the destroy on antigen என்ன ஆகும்னு கேட்டீங்கன்னா, which will be eliminated out of the cell. இந்த complete process நம்ம என்ன சொல்றோம்னா, we call it as a phagocytosis. WBC ல நீங்க இந்த क्वेश्चन உங்களுக்கு டவுட் வந்திருக்கு ஏன்னு கேட்டீங்கன்னா, மேபி ஆர்பிசி இல்லன்னு சொல்றதுனால you might have had the doubt WBC ல இருக்குமா, கண்டிப்பா WBC ல இருக்கும். ஏன்னா WBC வந்து phagocytic in functions, especially for the monocytes, neutrophils, இதெல்லாம் பாத்தீங்கன்னா phagocytic mechanisms. ஏன்னா அதிகமா வெளியில இருந்து வர செல்ஸ் எல்லாம், ஆன்டிஜென்ஸ் எல்லாம், பாக்டீரியா, வைரஸ் இதெல்லாம் உள்ள வரும்போது the defense mechanism is functioning, your cell mediated immunity எப்படி பெர்ஃபார்ம் ஆகுதுன்னா இந்த மெக்கானிசம் மூலியமா தான், by means of phagocytosis. அப்போ என்ன ஸ்ட்ரக்சர் ஃபார்ம் ஆகுது? ஃபர்ஸ்ட் யூ ஹேவ் a lysosome, the lysosome which fused with the bacteria and form phagosome, and phagosome and lysosome which again fused together and form a phagolysosome. இந்த phagolysosome என்ன பண்ணும்னா அதுல இருக்க லைசோசைம் உடைய அந்த என்சைம் வச்சு பாக்டீரியா வந்து டிஸ்ட்ராய் பண்ணிடும். அப்போ கில் ஆன பாக்டீரியல் காம்போனன்ட்ஸ் என்ன ஆகும்? which will be destroyed out of the, eliminated out of the cell. இந்த கம்ப்ளீட்டா ஒரு செல்லையே என்கல்ஃப் பண்ணுது, சாப்பிடுது அப்படின்னா this process called as phagocytosis. இதே வந்து ஒரு பாக்டீரியால இருந்து வந்து ஒரு சின்ன ஒரு ஃபிளஜலாவோ, ஒரு பைலியோ, இல்ல ஏதோ ஒரு கிளைகோ புரோட்டீனோ, இல்ல செல்வரோட காம்போனன்ட்டோ அது மட்டும் உள்ள போய் டிஸ்ட்ராய் பண்ணுச்சுன்னா that is called as pinocytosis. நேத்து நம்ம படிச்சோம் bulk transport mechanism where we discussed in the plasma membrane which performs exocytosis and endocytosis. In the exocytosis la வந்து which is going out, endocytosis la வந்து we discussed about phagocytosis and pinocytosis. Phagocytosis and pinocytosis. Phagocytosis is a cell eating mechanism and pinocytosis is a cell drinking mechanisms by which what happens? Why I'm telling this for you is this is for the reference like the lysosome which present in your WBC, because WBC is we're talking as a defense mechanism. How it performing, especially the cell mediated immunity. The cell is destroying the antigen, right? This is because of this particular cells. They are also present Kupffer cells, of course, Kupffer cells, alveolar cells, microglial cells. In the எல்லா செல்ஸ்லயுமே defense mechanism, defense cells. இந்த எல்லா defense cellsலயுமே வந்து Kupffer cells எங்க இருக்கும்? Where you could see the Kupffer cells which present in the liver, alveolar cells which present in the your lungs, and microglial cells present in your brain. இதெல்லாமே வந்து phagocytic cells நம்ம சொல்றோம். ஏன் இந்த phagocytic, macrophages phagocyticனா because this performs a function. அப்பனா அதுல என்ன இருக்குன்னு அர்த்தம்? The lysosome or lysozymeன்ற பாக்டீரியா இருக்குன்றதுனால தான் this can performs. அப்படின்னா நம்ம சாப்பிடுற சாப்பாடுலயோ இல்ல நம்ம சுவாசிக்கிற காத்துலயோ, so some way like somehow like the organisms getting inside. So then why we're not getting disease or we're not getting infected with that? Because these cells which performing the defense mechanism properly by means of phagocytic mechanisms, that is cell mediated immunity is getting established. So I hope I answered for this question. So now the time is 3:35. Right, so I'll give a next 5 minutes break for you. So after 5 minutes, so guys don't take much of time. So sharp 3:41 we'll meet again. So we continue with this chapter. Have a short break. I'll meet you again. Thank you. Welcome back, students. So we'll continue. So lysosomes that we discussed the last class, and the main function of lysosome is intracellular digestion, that is phagocytosis, that we discussed. Intracellular digestion by phagocytosis mechanism. Okay, fine. So I can ask me what's the main function of lysosome? It's intracellular digestion, the defense mechanism. So whatever the thing that we discussed in the previous part, all these things we make it as like it's a function. Okay. So next we'll discuss. We're done with the lysosomes. We'll start with the vacuoles. We'll start with the vacuoles. So the vacuoles present both in a plant and an animal cell, but the plant cell is a very prominent. The size of the vacuoles is it's definitely it's a big in size, so it occupies the central region. And whereas in animal cells, most of the time the vacuoles are not present. Maybe if at all present, like you can see like very small vacuoles that also is noticed in the animal cells, but the prominent vacuoles may not be seen. But in animal cells, it's not seen. But in plant cells, you could see the prominent vacuoles which occupies the center. The majority of the cell itself has been occupied by vacuoles in a plant cells. Fine. We'll start with the vacuoles. Vacuoles which occupies the 90% of the total volume of the cell. Total volume of the cell. Usually this rule applicable in case of plants actually. In plants only this rule applicable. So 90% of total volume that's occupied by the vacuole. And the layer, for example, you could see there's a cell. I'm talking if it's a like plant cell. The plant cell is completely occupied by the vacuole. And here you could see the nucleus. This is the nucleus part, and this we call it as a vacuole. Okay. And the layer which covers the vacuole. What is the name of the layer? Even we discussed yesterday, right? So what is the layer? The membrane which covers the vacuole. So what is the name of this particular layer? We give a specific term for this layer or the membrane which covers the vacuole. What is the name of this? It's called as tonoplast. The tonoplast is the membrane which covers the vacuole. Fine. Then there are types of vacuoles. What are the different types of vacuole? We have a sap vacuole, contractile vacuole. We have a contractile vacuole. We have food vacuole, and also we have a gas vacuoles. These are the different types of vacuoles that you could see in the different types of cells. Sap vacuole, contractile vacuoles. Even the contractile vacuoles and the lower organisms, it perform the functions like osmoregulations. Where you could see contractile vacuoles. Sometimes the vacuoles which also performs like for food, reserving the food, and sometimes it helps in a buoyancy of a cell, which makes the cells to float even. For that, you could see the prominent gas vacuoles can also be seen. So next we move on to the microbodies. Next we discuss about microbodies. Next to the vacuole, we have a specific structure. This structure we call it as a microbodies. These microbodies which performs important function in oxidative reactions. In oxidative reaction. The oxidative reaction usually which performs by this microbodies. Now we'll discuss one by one the types of the microbodies. We have peroxisome. Peroxisome types of microbodies. Peroxisome, glycoxisome. Peroxisome. Peroxisome. We have glycoxisome, and we have lomosomes. Lomosomes and spherosomes. Spherosomes, and we have something called as lomosomes. These are all the different types of the microbodies. These microbodies which perform the functions, different functions, but main function is oxidative reaction. These microbodies we can call it as peroxisome, and we have spherosomes, and we have spherosomes, and we have a lomosomes. When coming to the first part, peroxisomes, which usually we could see in a C3 plants. You could see in a, it's very common in a C3 plants. All the vacuoles are present in both prokaryotes and eukaryotes. It depends actually. It depends upon the habitat. It depends upon the requirement. It depends on the adaptation. So as per the adaptation, if the particular organism seems to be barotolerant in nature, then that required vacuoles has to be there. So it's not compulsory like all the organisms of prokaryotes and eukaryotes, and definitely it should have. It depends upon the kind of adaptation and which environment these organisms are growing. Fine. Coming to the peroxisomes usually could see in a C3 plants. What is the microbodies? The microbodies are basically membrane-bound organelles. we can define this microbody as a membrane bound organelle. This performs an oxidative reaction. So peroxisome, this type of microbodies, often you could see in a C3 plants. We already discussed about the C3 plants and C4 plants and what way it can be differentiated. Fine. Coming to the next part, and for example, for C3 plants, we take like cotton, beans. These all comes to the category of a C3 plants where the bundle sheet may not be having the chloroplast. The chloroplast is not present, but in C4 plants, the bundle sheet cells do have the chloroplast. In that way, you can differentiate. And the first stable compound which forms is a 3-carbon compound in case of C3. So that's the reason we call it a C3 plants. So these peroxisomes, these are present in a C3 plants, which performs a very important role, and by producing enzymes like catalase, urease, oxidase. The enzymes which produced are like catalase, oxidase. We have the catalase, oxidase, and we have luciferase. These are the different types of enzymes which produced by this type of peroxisomes. You take urease even, oxidase, urease. These are very common enzymes which produced by peroxisomes, and this facilitates playing very important role in a C3 reaction, especially C3 plants, especially photosynthesis. Coming to the glycoxysome. As far as the glycoxysome is concerned, this is quite common in case of seed. This type of microbodies, it's quite common. It's common in case of seed. Spiriozymes, again, this is responsible for majority of the enzymes like catalase, peroxidase, this type of enzymes which can be produced by spiriosomes. Lomosomes are actually the connective bridge that you could see between the cell wall and the plasma membrane. This present between the cell wall and plasma membrane. So on the whole, these are all microbodies. Stonoplast is the outermost membrane of the vacuole. The membrane which surrounds the covering membrane of the vacuole is called a stonoplast. We discussed the microbodies. Microbodies are membrane-bound organelles. This performs oxidative reaction. And the 4 types of microbodies: peroxisome, glycoxysome, and we have spiriosomes, and we have lomosomes. So peroxisomes is present in C3 plants, glycoxysome in a seed part that you could see. In spiriosomes, it contains again majority of the enzymes, and lomosomes is a connective, this type of microbodies which present between your cell wall and cell membrane. So we're done with the microbodies as well. Okay. So I do agree, my writing on the board may not be clear, but that's the reason whenever I write something, and also I'm dictating it. Because you can at least hear to that. If you have any particular segment, particular area that you couldn't understand, you can ask me so that I can repeat it again. So that's the reason I'm writing. At the same time, I'm repeating that. I'm repeating the same thing, whatever I'm writing on a board, I'm repeating. So at least you can listen to it. If again still the doubts are prevailing, you can ask me. All right, fine. So peroxisome, glycoxysome, spiriosomes, and lomosomes. So these are the 4 types of microbody. Next to lomosome, it's present. So lomosomes is present between the cell wall and the plasma. This is what you're asking, right? The lomosomes, where the lomosomes are present? The lomosomes are present in between the cell wall and the plasma membrane. Spiriosomes are responsible for the production of majority of the enzyme So since it's a time-consuming process, I'm not writing in full form. So because you telling guys I'm going very little bit fast. Maybe sometimes you tell very fast because we have a lot of other chapters to cover. That is the reason, right? I'm going a little bit fast because we need to cover 11th as well as the 12th standard chapters for you. So that is the reason I'm going a little bit fast, and also writing a little bit fast to the board and using short forms, because if I write in full everything, again, it consumes all our time because time is precious on both the sides. So I hope you guys can understand. Okay, so because I'm writing CW and PM is like cell wall and the plasma membrane. So these are the short forms. I often use it on the board. Kindly bear with me. This again for your benefit. I'm going a little bit fast, both as well as in writing, as well as when I teach. The reason is not to make you guys uncomfortable. You guys should understand we're doing it because we have a lot of other chapters also to discuss. We have other plant physiology and 12th standard chapters that we need to cover. Having this in your mind only, I am going little bit fast. If there is any discomfort that you are facing in your side, you can have a rewind, or you can add your doubts in your comment box, and you can approach me so that I can clarify your doubts. So I hope you can understand, right? Even I like to teach slower, but if I teach slow, like we cannot complete, the portions like as per the schedule. Thank you so much. Thanks for your understanding. So these are all micro body. Clear with this micro body. Then we'll start with the cell inclusions. Next, we'll start with the cell inclusions. The cells also having some inclusions. Cells having something called as inclusions. Some inclusions and foldings that you could see inside the cell, right? So as far as cell inclusions are concerned, some types of inclusions we call it as starch granules that you could see inside the cell. Most of the plant cells that you could see the starch granule. In animal cells, you could see mostly the glycogen granules inclusions that you could see. Then sometimes you could see the fat droplets. The inclusions may be in the form of a fat. These fat droplets can also be seen. Then you can also see aileron and crystals. Sometimes you could see this inclusion as a aileron, and sometimes you could see as a crystals. You could see as a crystal. So starch granules, where you could see this type of starch granules, often you could see is because the reserve food in the plant is a starch. So plant cells are the best example for this type of granules. Glycogen you could see in mostly in the animals, especially in the smooth endoplasmic reticulum. Smooth endoplasmic reticulum, and also you could see in case of liver, and also you could see in the muscle, and as fungi is concerned, you could see in a mold, the fungi, because fungi are two forms, mold form and yeast form. In a mold form, you could see even the presence of glycogen. Okay, so we try to complete this class by 4:30. Okay, as per the schedule. Then fat droplets you could see in the endosperm of castor. Endosperm, you would have studied about the endosperm, right? Endosperm of castor, the seeds, that is endosperm of castor and coconut. Can someone tell me what is the ploidy of endosperm? Haploid, diploid, triploid. So what is the ploidy? What is the ploidy of endosperm? If you were to study the endosperm, let's just have a quick recap of what are things that we discussed in the plant, the pollination, and the embryo formation, that zygote, the 7 cell 8 nucleate stage, and all. If you discuss, the ploidy of endosperm is 3N. 3N is the ploidy. So often they ask the question, and is it the same both in angiosperms and gymnosperm? The triploid, that is like, it's not polyploidy, it's a triploid. Is it same? The triploid is same both in angiosperm and gymnosperm? Yes or no? Or is there any difference? Of course, in case of you take angiosperm it is triploid. How about gymnosperm? Gymnosperm is it also a triploid? Can you justify? So if the questions they're asking the endosperm, in general they ask the endosperm, triploid is a correct answer. Please ensure the triploid, the question is for angiosperm or gymnosperm. Angiosperm, flowering plants, the endosperm, especially in case of coconut, you take in castor, the ploidy is 3N. But in case of gymnosperm, what is the ploidy? Ploidy of endosperm in gymnosperm plants, like Cycas pinus, that you could see. What is the ploidy of the endosperm? Can someone tell me? If it is not a triploid, no, its answer is no. Then what is a ploidy? It is haploid or diploid? Definitely it's not a triploid. It's haploid or diploid. What is the ploidy of endosperm in case of gymnosperm? Okay. Aileron. So where did I study about the aileron? It is haploid, exactly. In case of gymnosperm, it is haploid in nature. So you need to be very careful in reading the question. They're talking about endosperms. We be very enthusiastic in answering the correct answer, giving the correct answer. You go for triploid. But please understand whether they're asking a gymnosperm or angiosperm. If they're more specific, angiosperms, endosperms are, it's triploid. But in terms of gymnosperm, it is haploid. N. So this fat droplets, you could see the endosperm in case of castor and the coconut. You've studied about the coconut, like what the water that you're drinking, right? That is what இளநீர் சொல்றோம்ல, இளநீர் குடிக்கிறீங்க, இளநீர் குடிச்சா வழுக்கிய சாப்பிடுறீங்களா? So what is that actually? It's a endosperm. அதுலயே ரெண்டு type of endosperm இருக்கு, cellular endosperm and the nuclear endosperm. There are two types of endosperms that you could see. இது again எதுல வருதுன்னு கேட்டீங்கன்னா, it's again the fat droplets only. Coming to the aileron. Aileron நீங்க எங்க படிச்சிருப்பீங்கன்னு கேட்டீங்கன்னா, wheat grains seedல படிப்பீங்க. Wheat grain. So in the wheat grain, the monocotyledon seedsல நீங்க படிச்சிருப்பீங்க. So Inclusions na சுழண்டு இருக்கிறது இந்த மாதிரி. For example, if you're talking about the cell, செல்லுக்குள்ள இந்த மாதிரி அங்க கிளம்பா இருக்கும், இந்த மாதிரி குரூப் வச்சு கிளம்பிங் மாதிரி இருக்கும், இன்க்ளூட் அப்படியே கிளம்பா இருக்கும். ஸோ ஒரு இடத்துல ஸோ அந்த இன்க்ளூஷன்ஸ் தான் என்னன்னு கேட்டீங்கன்னா இந்த மாதிரி இருக்கும். அது ஃபேட்டா இருக்கலாம், கிளைகோஜனா இருக்கலாம், இல்ல அலிரோனா இருக்கலாம். ஸோ this is what the inclusions mean. In aliron you could see the wheat grains, you could see this type of things, and kind of like crystals. There are certain chemical compounds, the chemical compounds like oxalate, the chemical substance which you could see. Even the chemicals can be also act like a inclusions. ஒரு இடத்துல வந்து PHP granules சொல்லுவாங்க. I don't know how many of you heard about it. Polyhydroxy butyrate ன்னு சொல்லுவாங்க. This PHP granules கூட ஒரு type of granules. Can someone tell me the PHP granules are cyanophycean granules, this type of granules where this is present? This is prominent over at you would have studied the PHP granules, polyhydroxy butyrate granules, cyanophycean granules. இந்த டைப் ஆஃப் கிரானுவல்ஸ்ல நீங்க எங்க படிச்சிருப்பீங்க? இது பாக்டீரியால படிச்சீங்களா? எந்த ஏரியால படிச்சிருக்கீங்க? ப்ரோகாரியோட்ஸ்ல இருக்கிறோமா இல்ல யூகாரியோட்? there are particular area you would have studied about PHP granules, polyhydroxy butyrate granules. This is also kind of the chemical compounds which comes like specific type of what is it called, organisms like bacteria. You have the polyhydroxy butyrate granules. Okay, I humble request guys, please have the relevant chats here. So my request is like, don't distract the other students who are listening to the class because they are listening to the session. So don't post anything like which acts like a distraction for the others. Thank you for understanding. So polyhydroxy butyrate granules, which bacteria has got this type of granules? Next we move on to the special structure of the cell. Coming to special structure, apart from this we have some special structure of the cell. Come to the special structure of the cell. Talking of the special structure of the cell, we have centrosome. Centrosome is one of the special structures. Centrosome. Okay. Coming to centrosome, cytoskeleton, cilia and flagella. Centrosome, cytoskeleton. Then we have cilia and flagella. These are the other important, the special structures that you could see in a cell. Coming to the centrosome, this was proposed by Boveri. The name of the person, Boveri, in the year 1888. He proposed the centrosome structure. These centrosomes, what I studied about centrosomes. These centrosomes are responsible for what? Responsible for the spindle fiber formation, ester formation which takes place, especially in the cell division. Coming to centrosome, what the centrosome which consists of? Centrosome consists of a structure called as diplosome. So diplosome consists of two rod shape structure, and two cylindrical structure. Cylindrical, DRICL, cylindrical structure, two rod shape structure and two cylindrical structure. And these two rod shape and two cylindrical structures, and this cylindrical we call it as a centrioles. This we call it as a centriole. This two cylindrical part we call it as a centrioles. And these diplosomes, right? The diplosomes and two rods and the two cylindrical. So one will be acting like a peripheral, another will be acting like a central part. So together, this consists of fibrils and hub. This consists of fibrils and hub region. This fibrils and hub, okay, this is responsible for a cell division, and this starting developing a radiating threads, and this radiating threads we call it as an aster. The cell division நடக்கும்போது பாப்பீங்க, the kinetochore, the cell is getting separated. I mean the cystochromatid is getting pulled up on both the sides to near to the poles from the equator. இது எப்படி புல்லா எப்படி யாரால the process, how this process is happening? This is because of what? This is because of this process, that is by means of this centrosome, especially by means of centrioles. The centrioles which consist of fibrils and hub. This fibrils and hub, these two are playing very important role in what? In the cell division, where it the centrioles develop radiating threads. These radiating threads are called as aster. The radiating threads. These radiating threads are in the area is very important, because when the next part, if you start studying, like we're discussing about, like, mitosis, meiosis படிக்கும் பொழுது the spindle fibers பத்தி படிப்பீங்க. அப்ப the spindle fibers the centrioles and the structure will be studying. So which special structure is responsible, where you could see this centrosome is called a diplosome. This diplosome which consists of two rods and the two cylindrical parts. The two cylindrical parts which consist of centrioles. This centrioles which consist of the fibrils and the hub region. This fibrils and hub region, these two regions are responsible for the cell division. During cell division, it develops a radiating thread-like structure. For example, your cell எடுத்துட்டீங்கன்னா இங்க தான் சென்ட்ரோசோம் இருக்கும். நீங்க you would have noticed இந்த மாதிரி radiating threads மாதிரி வரும். இங்க பாத்தீங்கன்னா இந்த மாதிரி radiating threads மாதிரி you would have seen the diagram. The radiating threads இங்க வந்து உங்களுக்கு வந்து chromosomes இருக்கும். Central part வந்து you would have seen the chromosomes here, right? இந்த radiating threads இருக்குல்ல, the radiating threads only we call it as ester. This part is called as centriole. This is called as centriole. I hope. Okay. So yes, this present, and one more thing, this is present only in an animal cell. Plant cells la வந்து இந்த centriole formation irukad. That also very important, right? Centrioles which present only in the animal cell, which is not present in the plant cell. So thanks for reminding that. Clear. So other understanding எப்படி structure வருதுன்னா, diplosome two rod structure, two cylindrical structure called as centrioles. These two centrioles that we call it as, we classify into fibrils and hub. This together which forms a radiating structure. This radiating structure, சொல்றேன். This radiating structure we call it as aster, and this playing very important role at the time of cell division. Next chapter, I mean cell cycle வரும்போது இத நம்ம பாப்போம். Actually structure மட்டும் பாப்போம். அதோடைய introduction வந்து இந்த சாப்டர்ல டிஸ்கஸ் பண்ணிட்டோம். So cellல வந்து you do have a special structure called as centrosome, and centrioles which originate from the centrosome. So this is the first part, and second part we go with the next part, the special structure. This is the cytoskeleton. So the cytoskeleton, which is otherwise called filament or tubules, and this consists of microtubules, microfilament. This consists of microtubules, microfilament, and we have intermediate filament. Okay, so cytoskeleton or the filament. Here we have microtubules, microfilament, and we have intermediate filament. When come to the microtubules, the size is 25 nanometer. 25 nanometer is the size of the microtubules. Then we have microtubules. Microfilament is 7 nanometer. Intermediate filament size of 10 nanometer. It's a 10 nanometer. So we are discussing about the special structures of the cell. When we come to the special structure of the cell, centrosome that we discussed, centrosome is responsible for centriole. The centriole is responsible for the fibrils, and this eventually which forms for the thread-like, radiating thread-like formation. This we call it as aster. Okay. The second part, we start with the cytoskeleton. The cytoskeleton is otherwise called as filament, and this consists of three regions: microtubules, microfilaments, and intermediate filament. Here, the size of microtubules is 25 nanometer, microfilament is 7 nanometer, and intermediate filament is 10 nanometer. Right. Coming to the next part, cilia and the flagella. This cilia and flagella, which consists of basal body, hook, and filament. This consists of three parts: basal body, hook, and a filament. Now we'll see the diagrammatic representation of cilia, basal body, hook, and filament. That we'll see. One second. So this is about this image, which tells you about the cilia and the flagella. As for cilia and flagella, you could see we have the plasma membrane and we have the radial spoke. I told you like three important regions that you could see, right? What are three regions? Basal body, hook, and filament. This consists of basal body, hook, and a filament. Basal body, hook, and we have a filament. Either one where you could see is in case of flagella, and also you could see in case of like pili and a fimbriae. These are the structures that you could see in case of bacteria. Bacterial, you could see the difference. Is there any difference between flagella, fimbriae, pili? These all structures that you could see in case of bacterial cell. So in the bacterial cell, you could see three types of the locomotory like organs, that is flagella, pili, and a fimbriae. All this flagella, pili, and fimbriae, all these are playing very important role in this mechanisms, like which has got basal body, hook, and a filament. So how actually this arrangement is actually 9 plus 2 array. This arrangement is in the form of 9 plus 2 array. So for example, if you count the 9 plus 2, 1, 2, 3, 4, 5, 6, 7, 8, 9. So this gives 9 in number. Here, this gives 9 in number. These are 9 in number. And here in the central part, you could see the central microtubule has got 2. So this gives 9 plus 2 array. This we could see in case of fimbriae, pili, flagella, but each has got different functions. Fimbriae is for what is the main function? It's attachment actually. Fimbriae is for the attachment. Pili which plays an important role for conjugation. So you study like conjugation there. F plus F minus there you study. So this is for conjugation. So this is for conjugation. The pili is playing very important role. Flagella is for the locomotion. So fimbriae is for adherence or for the attachment. So it is like lotic habitat, right? So how these microorganisms which get adhere onto the rocks, the sticky surfaces, this by means of fimbriae. Pili is for the conjugation. Array means the arrangement. Array, we're talking about the arrangement, how this getting arranged. So these 9 tubules which arrange the outer part, and this 2 at the center. So the array we're talking about 9 plus 2 array. So 9 in the outer circle and 2 in the middle part. So this gives a 9 plus 2 array. So I hope you're clear with this microtubules. From microtubules they ask about 9 plus 2 arrangement. This is quite common with all the locomotory surfaces with the flagella, pili, and the fimbriae. So we're done with the special structure of the cell. And at last part of this chapter, we're going to discuss about microscopy. So yeah, it's having central hub. This 2 array is there, right? The 2 array is actually the central hub. It's a 9 plus 2 array. So it's a central hub. It's the center. Coming to the next part, microscopy. Can someone tell me who is the father of microscope? We'll start with the microscopy. Who first found the microscopy? First microscopy was found. Who's the father of microscope? We have many scientists they came after that, advanced types of microscopy they are found. But in spite of that, the person who first identified or discovered the microscope, the concept of microscope. So who's the father of microscope? Coming to the type of microscopes, we'll discuss. Here I classified the main types of microscope, light microscope. Here we have the transmission electron microscope. This we call it as a TEM. Simply we call it as TEM, which stands for transmission electron microscope. And this is the scanning electron microscope. Of course, I'll be giving the classification of microscope. Who is the father of microscope? No, it's not Robert. It's not Jason. It's not Jason. No. So we'll close by 4:30. 4:30, we'll wind up. Coming to the transmission and scanning electron microscopes. This is very important part, the difference between SEM. SEM is a scanning electron microscope and TEM is a transmission electron microscope. Sometimes the question comes like which types of microscope which can give 3D image. 3D image is by the scanning electron microscope. The scanning electron microscope is quite common where we can get the 3D image. The father of microscope is Anton van Leeuwenhoek. Anton van Leeuwenhoek is the father of microscope. Initially, he is the person who was first called as father of microbiology, but later part only Louis Pasteur, other guys like contributed. So the credit, the father of microscopy is the Anton van Leeuwenhoek. Fine. So scanning electron microscope, 3D images, and it is for beam formation. So a scanning electron microscope works on the principle of beam. It works on the beam formation. And here the transmission electron microscope is a direct imaging. It's a direct imaging. Coming to the magnification scan electron microscope, its magnification is an important feature of microscopy. It's a 2 million. Here it's approximately, it comes like 50 million in case of transmission electron microscope. Then Here it scans larger areas, the scanning electron microscope. Here the scan only the thin samples. And here the limitations of scanning electron microscope is conducting samples and the charging effects. Here limitations is a magnetic sample. So mostly you get a questions from this part, the scanning and transmission electron microscope with the magnification. Here the magnification is, it's like 2 million. Here the magnification is 50 million. And this which type of electron microscope which gives a 3D image? The scanning electron microscope which gives a 3D image. Okay. See, we have different types of microscopes. We have fluorescent microscope, phase contrast microscopes, right? So the Jasons, these people, like, they found like later the inventions, but the concept, the foundation of the microscopy, which is given only by Anton van Leeuwenhoek only. So we discussed. So this is about the scanning and the transmission electron microscope. So coming to the main classification of microscopes. So coming to the classification of microscopes. These microscopes are generally classified into three segments. What are the three segments of the microscope? The three segments: light microscope, electron microscope, and we have the advanced type of microscopy that is called a scan probe microscope. Scan probe microscope. Coming to the light microscope, you would have used in a school's compound microscope. The compound microscope, which comes under the light microscope. Then we have phase contrast microscope. Phase contrast microscope. We have fluorescent microscope. Fluorescent microscope. And also we have the other type of microscope. The other type of microscope is DIC. DIC is differential interference. DIC microscope is a differential interference contrast. Differential interference contrast. And the other type of the light microscope, we call it the fluorescent microscope. We have a confocal microscope. Confocal microscope. Coming to the electron microscope, transmission electron microscope and scanning electron microscope. In the last slide we discussed about the transmission and the scanning electron microscope. Scan probe microscope, scanning tunneling microscope. Scanning tunneling microscope. Tunneling microscope. Then atomic force microscope. Atomic force microscope. These are the broad classification of a microscope. Okay. When you come to this is a normal microscope, the compound microscope. You would have noticed, like, we have the magnification at 10x. Often we use a 10x, 45x, and its magnification is like 100x. From this part of the microscope, you either get a questions from the transmission and scanning electron microscope, or you get a question from the resolving part. Resolving part or magnification. These two areas, like, they ask which among the following is the correct statement, which among the following is the incorrect statement, right? DIC stands for differential interference contrast. Differential interference contrast is the DIC. Okay, so compound microscope. Okay, fine. This is the common microscope that you would have seen in the school itself, the compound microscope, where you could go for staining and everything, where you cannot see the virus and all in the common microscope. Okay, then phase contrast microscope. Coming to the phase contrast microscope. So what might be the main application of the phase contrast microscope that we'll see, and finally we move on to the next part. As for the light microscope, this can be classified into the bright field and the dark field microscopes. What makes the difference between the bright field and the dark field? As for the bright field is concerned, the background is bright, where the specimen that you could see is a dark actually. So that is a bright field. But in dark field microscope, the dark field, I mean the background is actually the dark, but the specimen, that organism that you're looking at it is actually, it's bright. This is called a dark field microscope. So this makes a difference between the bright field. The bright means the field is, the background is bright, then it is a bright field. The dark field microscope, where the background is dark, then we call it as a dark field microscope. I told you in the last slide, it's a transmission electron microscope, TEM. SEM stands for scanning electron microscope. And DIC is a differential interface contrast. Okay. So phase contrast, I just give the applications like where we use like this type of microscope. Compound microscope we commonly use for like looking at the fungi, bacteria, the staining specimens and everything. Coming to phase contrast microscope, here we have the special type of condenser that you'll use actually. The special condenser with the annular diaphragm, that we use in the phase contrast microscope. This is actually the principle behind where you could see the direct and the reflected rays. Brought together. Direct and reflected rays. These two brought together in case of phase contrast microscope. Fluorescent microscope. Can someone tell me what is the application of fluorescent microscope? And before that phase contrast microscope, we use a special type of, the condenser that you use. What is the name of the condenser? Fritz-Zernick condenser. Fritz-Zernick condenser. Is a special type of condenser that we use in case of phase contrast microscope. This Fritz-Zernick condenser, which helps to combine the direct and the reflected rays together. So this is the principle behind the phase contrast microscope. This is because of the special type of condenser called as Fritz-Zernick condenser. Coming to the fluorescent microscope. So when you talk about fluorescent microscope, this is used for usually like identifying the microbes in a tissue. You want to look at the microbes in a tissue. You want to use the microbes in a tissue, you require ultraviolet rays. Here it requires ultraviolet rays, and you require a special type of staining that is called as acridin orange. You require acridin orange. It's actually a special type of dye that you require, right? This is called as fluorescent microscope. Since you guys may think this is all like an additional things that I'm discussing about, maybe you get one question from the microscopy. So the outline that I'm giving, right? So for that, you please listen to it. Fluorescent microscope, the application where you could see the microbes in a tissue, where this specimens which is get exposed to the ultraviolet rays, and the specimen supposed to be stained with the acridin orange. Coming to the, for example, if I ask you organisms like spirochetes, like Leptospira, Treponema pallidum, which causes syphilis. For this type of organisms, like for this type of identification of this particular bacteria, which type of microscope that you prefer? I hope you got the question. For identification of the organisms like spirochetes, like what are the spirochetes? Spirochetes are the organisms which present like this, the bacteria which present like this. Is this type of snake-like structure, serpent-like structure, we call it as spirochetes. Example for spirochetes, Treponema. Treponema is actually the causative agent of syphilis. To identify this type of bacteria, which type of microscope that you prefer? Which type of microscope usually you prefer for the identification of the spirochetes like Treponema pallidum or Leptospira? All these things, which type of organism that you prefer? Fine. So the fluorescent microscope is actually the microbes that you want to like see directly from the tissues, where you go for the ultraviolet rays and it has to be stained with the acridin orange. This you can do it. Okay. Then we have the confocal microscope. Confocal microscope, this is for the 2D and 3D images. You want to get 2D and 3D images with the light microscope itself, you can go with the confocal microscope. Okay. Here you can use a laser light. This prefer the laser light. The laser light is required as far as your confocal microscope is concerned. And coming to DIC also, like you can use a beam of light, and which is by the prism. Here we use a beam of light, which is getting scattered through the prism. And this is also, by this also you can see the 3D images. So these are the main types of the microscopes as a light microscope. So light microscope, compound microscope, phase contrast microscope, fluorescent microscope, differential interface contrast microscope. Then we discussed about the confocal microscope. In the last slide, we discussed about the electron microscope, where the magnification is 1,000, 10,000 times it is more. That is transmission electron microscope and scanning electron microscope. At last, we have a scan probe microscope. Coming to scan probe microscope, we discussed about the scanning tunneling microscope and the atomic force microscope. This is quite expensive microscope. Where do we use this type of scanning tunneling and atomic force microscope? Where you want to see the molecules which present inside the cell. Inside the cell, if you want to see exactly like the molecules, the DNA, RNA, macromolecules, micromolecules, DNA structures, and more deeper the molecular structure, molecular details that you want to study, then you can go with the scan probe microscope. Atomic force means you can even go with the atoms. You want to study about the atoms which present in the cell, right? The atoms which present in the cells, like you want to study detailed picture about the atoms and in deeper. Then you go with the atomic force. You want to go with the molecular study, the micromolecules, macromolecules, the DNA, the RNA, and everything. Then you go with the scanning tunneling microscope. These are like advanced microscopes, and it is also like expensive way, right? So coming to DIC, where you're using the microscope, the based on the principle is like you're using the beam of light, and the beam of light which has been passed through the prism. So here no staining required. And one more thing, in DIC, you need not prefer any staining. Live, you can see. So there's no need of staining. So do not worry about the staining and everything in case of the differential. It's not depression. Differential interference contrast. Differential interference. It's not depression. Differential interference contrast. So no staining is required for DIC. You can see the 3D images. The principle behind DIC, where you're passing the beam of light. This beam of light is passing through the prism, so that you will be getting the images. So by dark field microscope, first of all, is exactly like I told you for spirochetes, which type of microscope is preferred? Dark field microscope, where the organisms which will be like light, where the background is dark. For spirochetes like Leptospira, Treponema pallidum, Borrelia, these are all like spirochetes, which comes in spirochetes family, which will be like snake-like organisms. For confirming this type of microscope, sorry, for this type of organisms, we prefer dark field microscope, where the background is dark and the organism, the specimen seems to be bright. So what type of condenser? You need not, you have a question like, is there any this special type of microscope called as dark field microscope? No. Any point of time, the bright field microscope can be changed to a dark field microscope. Then what you have to do? You have to change only the condenser. You change the condenser, that is like the diaphragm. If you change, what type of condenser that you need to incorporate to change light microscope to the dark field microscope? The type of condenser is a Is condenser. This type of condenser that you can incorporate, you can insert, so that what will happen? The bright field microscope will become a dark field microscope. So there is no separate dark field microscope. Only thing that you're gonna change is the condenser. What type of condenser? You use the same bright field microscope, but you just remove the condenser that is existing in a bright field microscope. You incorporate this condenser, a Is condenser, automatically your background will be dark and your specimens will be bright in nature. Then it will become a dark field microscopes. When you have to use, you want to use when you need a you want to identify the organisms like spirochetes, like Treponema, Borrelia, Leptospira. For these kind of things only you can go for this type of things. I think it's only the last 5 minutes more. I just conclude with the microscopy. So as far as the microscopy has got over, then we move to the microtome. Microtome, what is a microtome? Can someone tell me what is microtome? Microtome is a short blade. Microtome. What is microtome? Microtome, where you are going with a sharp blade, like meat cutter. So how we chop the meats into small like pieces. The same way, like how when you go for like slide preparation or something, the microtome is a place where you go for like thin sections of your samples. That's in thin samples that you can get it, and then you go and visualize the stain it and look it on the microscope. So microtome is an instrument. It's got a sharp metal blade for the thin sections. This used for the both light as well as the electron microscope. Then go for cell fractionation. Then we have technique called as cell fractionation. Fractionation. So what do you mean by cell fractionation? You want to identify any part of the cells, the DNA, RNA. First, the cell has to be bursted. So how it can be done? The cell fractionation is used for isolation purpose. You can isolate, and then you can go for centrifugation. You can go for a centrifugation technique. So how we can isolate it? You can isolate by just placing the particular cell, the solution in a sucrose solution, and then you just keep it in a deep freezing conditions. You keep it in deep freezing conditions like 0 to 0.4 degree centigrade. Sucrose solution, keep it in a sucrose solution. This is of like 0.2 molar 0.2 molar sucrose solution, then you just keep it in the 0.4 degrees centigrade. Automatically what will happen? The cell will get bursted. So the inner components, DNA can be isolated. So then, or you can go for centrifugation. Based on the density gradient, you can sediment it and then you take the fraction of that cell and you can go for the technique. So this is called as cell fractionization. And we have chromatography. We have chromatography. So can someone tell me what are the types of chromatography? What are the types of chromatography? We have column chromatography, partition chromatography, gas chromatography, affinity chromatography, then ion exchange chromatography. Ion exchange. You would have studied about the paper chromatography. Yeah, it's a common technique that you use in nitrogen solution that you do the paper chromatography. Yeah. So these are the common chromatography. What is chromatography? This chromatography is a principle of proportion coefficient. So used in isolation and purification of materials. Used in isolation. So if you want to go for isolation and purification of materials. Isolation and purification. So isolation and purification of materials. Isolation purification materials, you can use a chromatographic technique, column partition gas chromatography, affinity chromatography, and we have the ion exchange chromatography. So I show the diagram. This is actually diagrammatic representation. We have the liquid chromatography. This image which represents your liquid chromatography. So one will be in stationary phase, the other one phase will be in the liquid mobile phase. Gas chromatography you take, this is the polarity you could see the gas mobile phase, and the other one will be the liquid stationary phase. So planar chromatography, one will be in mobile phase, another one will be in the stationary phase. So these are like additional information that are given for you, the chromatography. Along with this, we have the staining technique, the cytological staining technique, the nuclear staining technique, so that cells can be identified. Clear. Then we have a technique called spectrophotometry. So what is spectrophotometry? Spectrophotometry. Spectrophotometry. And this is actually X-ray crystallography. X-ray crystallography. Crystallography. Guys, these are all only additional information for you. This is not the part of the textbooks. So X-ray crystallography, where the X-ray beam which get passed on crystallized structure and the rays will be get diffracted and the film, it will be get reflected onto the film, then you can identify the image. Then also we have something called as spectrometry technique. Spectrometry you would have studied in the spectroscopy, like by means of calculating the optical density. You can identify what is actually the volume of like molecules which present. The spectrometry technique also that you can follow. And we have the flow cytometry. So flow cytometry is again the technique. So why I'm telling all these things are like, these are the different terms like of advanced phases, right? Where you be using X-ray crystallography, spectroscopy, flow cytometry, all this technique that is quite common. Like most of the cases nowadays, the robotic labs where they're using this type of techniques. No longer they use like compound microscope, very old techniques now. Now the external crystallography, auto-radiobiography, by using radioactive isotopes, only they're incorporating. So this, need not worry about the last part of this chapter, because it's all like only additional information that are given for you. So with this, these are the tools that we are using: X-ray crystallography, spectroscopy, flow cytometry. These are the different tools by which we can identify. You want to identify the inner part of the cells like molecules. These are advanced parts that you can always remember. So if any questions comes in flow cytometry and everything, you need not worry like I didn't hear about all those things. At least if time permits, like if any questions related to flow cytometry comes, because I don't want to make this class so boring, because it's maybe many of you guys like first time that you're hearing all these things. So I don't want to bore you guys again by teaching all the things which not given in the textbook, but understand these are the advanced techniques that you're supposed to learn, right? Any advanced techniques like flow cytometry and the spectroscopy, or how to calculate the optical density and everything, that time you should not think like I didn't study anything. Maybe it may not given a textbook, doesn't mean like you have to limit yourself. You can explore additional information about the cell, because these are all the things which related with the cell. So I really appreciate that you guys even though the last part of the session is a little bit boring for you, so you patiently listen to this class. And tomorrow, right, we'll start with the next chapter, that is cell cycle, mitosis and meiosis, that we'll discuss. And I hope you guys got the received test papers. You just follow the test papers, and very shortly we'll be giving the answer key for you. So with that key, like you try to answer, and also if any doubts or queries, you please don't feel hesitate to contact us. So we are really happy to. Yes, I do agree. Like it's boring session the last. So because we crossed the stage for the students' life, because some things if it's not you're not aware of the part like that also like post session. If the thing is not aware for you, if it seems to be boring for you, understand one thing. I'm not dumping everything on your head, but you guys should understand you need to learn the additional information, so you're not stuck only with the textbooks. There are other techniques like extracrystallography, flow cytometry. At least you know what is all this concept is all about, so that it will be like good for you. Okay, so thank you so much guys. So I hope like tomorrow we'll have the interesting session with the cell cycle, and I try to not to make this class so boring once again. All right, thank you so much, and please be touch, you make use of time, and just try to go through with all the test papers that we're doing it and make use of time effectively. I really appreciate your time and patience. Thank you.